Good morning, Armula. Good morning. மியூட்ல இருக்கு மைக். மைக் மியூட்ல இருக்கு. டீன் மேம் ஜாயின் பண்ணதுக்கு அப்புறம் ஸ்டார்ட் பண்ணிக்கலாம். உங்களோட மைக் மியூட்ல இருக்கு ஆறுமுகம். மைக் மியூட்ல இருக்கு.
ஆன் பண்ணிடு. இப்போ ஆன் ஆயிடுச்சு.
ஓகே. டீன் மேம் ஜாயின் பண்ணிடுவாங்க. கொஞ்சம் பார்ட்டிசிபென்ட்ஸும் ஜாயின் பண்ணிட்டோம். அதுக்கப்புறம் ஸ்டார்ட்
சரிங்க.
ஒரு 5 மினிட்ஸ்.
சரி, ஓகே.
அதுக்குள்ள ஸ்லைடு வேணா ரெடி பண்ணி வச்சுக்கோங்க.
ஸ்லைடு எல்லாம் ரெடியா இருக்கு.
ஓகே.
அப்படியே வந்து போட்டு இது கொடுத்துட்றேன். ஷேர் பண்ணிடு.
ஓகே.
Good morning, sir.
டீன் மேம் ஜாயின் பண்ணிட்டாங்க. குட் மார்னிங் மேம். மேம் ஸ்டார்ட் பண்ணிக்கலாம்.
Good morning, good morning. Welcome, sir. We will
அன்மியூட்
start.
பண்ணி பேசுங்க. அன்மியூட் ஓகேங்க. ஸ்வர்ணா.
Good morning, one and all present here. Good morning, one and all present here. If you see the positive slides of everything, you will be able to live a much richer life than others. We, Raj Rajeshwari and Swarna Lakshmi, research scholars, Department of Botany, Avinashilingam Institute, Coimbatore, are privileged in mastering the ceremony.
A day without prayer is a day without blessing, and a life without prayer is a life without power. With this, we will begin this providential function with a prayer song. Now, I would like to invite Yangchen Dolmakom, research scholar, Department of Botany, Avinashilingam Institute for Home Science and Higher Education for Women, Coimbatore, to sing the prayer song.
रघुपति राघव राजा पतित पावन सीताराम रघुपति राघव राजा राम पतित पावन सीताराम भगवान ईश्वर तेरो नाम सबको सन्मति दे भगवान रघुपति राघव राजा राम पतित पावन सीताराम.
Coming together is a beginning. Keeping together is progress. Working together is success. It's my pleasure to welcome all the dignitaries, eminent speakers, deans, HODs, faculty members, research scholars, and students of various departments to this virtual program. Now, we'll begin the technical session by introducing the resource person, Dr. Arumugam, Professor. He has teaching experience of 18 years and research experience of 18 years. He has guided 2 PhDs, currently 6, and 24 MPhils, and 39 PhD students. PG students. He has attended several national and international conferences and published about 35 international and 3 papers in national journals. He completed projects funded by UGC and AURF with worth 12 lakhs, and currently he has Hello, ma'am, am I audible, ma'am? Ma'am, am I audible, ma'am? Hello.
Audible. Okay, audible.
Yes, sir. Yes, sir. He has he completed projects funded by UGC and AURF worth 12 lakhs, and currently he has an ongoing project worth of 15 lakhs in Dansky. He is also a member of Board of Studies for MSc and MPhil Botany courses in Alagappa University, member Board of Studies MSc Botany Alagappa University, affiliated colleges, chairman of Board of Valuation MSc and MPhil Botany in Alagappa University, chairperson Question Setting Board MSc Botany in Alagappa University, and representative Distance Education Examinations Alagappa University, member Inspection Squad Examinations of Alagappa University affiliated colleges, member Research Center Inspection Committee Alagappa University affiliated colleges member, Board of Studies for MSc Life Science Tamil Nadu Teachers Education University Chennai. Thank you, sir. Over to you, sir.
Hello, good morning. Hello.
Sir, you're audible, sir.
Audible. Okay, I start my lecture.
Yes, sir.
Okay, thank you for very nice introducing about me.
Thank you, sir.
And also I thank Avinashilingam University Registrar and Vice Chancellor, Dean Research and Department of Heads of Department of Botany, and my dear students for all all of good morning one and all. So my today my presentation polish.
Sir, please unmute your mic, sir.
அது தெரியும், பாதி இந்த பக்கம் தெரிய மாட்டேங்குது. ஹலோ, கேக்குதுங்களா? ஓகே ஓகே. ஹலோ, ஆடியபிள்?
Yes, sir.
Okay, okay. So today topic, I think past 3 days they are ongoing for a wonderful lecture department. So I thank for inviting me presentation my lecture, the gene in environment and future application. So what is gene? Where is in environment? How is possible in environmental gene? So there's also many questions rising for you. So most of the genes are regulated for the environment. So the genotype, the genotype and variation of the several genomes and interaction with the genetics. So the interaction genetic control of sensitivity of the environment. All the environmental factor that impact and directly or indirectly affecting for the organism and the changing for the phenotype and genotype. So many organisms changing, adapting and based on the environmental condition. So the environmental condition and the regulate the gene expression. The stable environment and a stable condition, gene expression is very good. If you unstable and environment and unstable condition, the gene expression level is altered. So you know about what is cell, the world within the entire world within the complex of cells, the cells and the cells entire life they are controlled. So out there androcynin, red and blue, so the different colors of the flowers, fruits. So that attracted the gene interactions. So this is a color androcynin. Another is got is carotenes. The carotene genes is present expression the carotene form. The carotene is like a and a bread trouble and the carotenoids. This is a yellow color. Another the green of the basic pigment. So you have to varicate it. The chlorophyll is the alteration. The gene is environmental gene, environmental interaction, alteration of chlorophyll synthesis. Some gene is directly are affecting for the chlorophyll synthesis in the where Mirabis jalaba already experiment done it and mentally in genetics. And the cyclic of the season, various season, various forming of the environment, the flower and making flower and the differentiation leaf, everything. The seasonal variation. The seasonal variation is regulating for the gene expression. So you have to see this wonder of the color. Whether you have to deficiency, if you deficiency or pathogen attack, and it's affecting for the gene regulation, gene control, and gene expression through the environment. So those are all in fact, and already the deficiency syndrome, everything you find out. Some genes are silencing, like RNA silencing and non-coding RNA. They are identified. The color variation identified. So if you RNA silencing means the RNA is single stranded. If you double stranded RNA, if you convert the double stranded and it is non-coding RNA. RNA anytime the single DNA sequence they are expressed and the silencing they are expressed. If you complementation of another one DNA complementation RNA, and that is called non-coding. That is stop the RNA sequence. That is called RNA interference. The RNA interference, most of the research paper published for blocking of the gene express or block knockout, gene knockout. There are so many applications they are using for RNA experiment. You have to see this color. Because of color variation is a man-made color. So this photosynthesis And these environmental conditions impact so many genes and the proteins and the protein modification, remodeling, and transcription factor. They are activate or inactivate because of light. The light, if you pass the proper light, they are given. They are chlorophyll and converting light energy to chemical energy like a glucose. So if you call alteration of light and like a cytochrome oxidase, cytochromes, cytochrome B6 and B20, these all cytochrome directly 60, 700 and 600 as that BR form and BFR form. So those BR and BR form because of alteration of the light receiving day. So this light receiving time or harvesting light, it is divided into the three categories: long day plant and short day plant and a medium day plant. So this all is regulating for light. That is, light also control of the gene expression. So you can see this wonderful leaf, the structure, and the leaves of parenchyma, sponge parenchyma, and it is all mad. These are miracle structures. The sharing sunlight, the leaves arrangement is the environmental impact, the alteration of the direction of the leaves and the harvesting 100% sunlight and the different angle of the leaves formations. Different, they naturally differentiate for the unique leaf structure to identify plant and if you summer and the plant leaf is very small and those leaves like a needle like a structure like a zero points. So this all is alternative naturally that regulate the genes and the particular environmental condition. So to identify the different leaf structure in the different area, the environmental factor, determination of the shape and the structure of the leaves. So arrangement of the plants also harvesting sunlight, different manners, and the plant automatically changing that, like that the movement of plant because of environmental impact. So the sensitivity of the communication, plant also communicated, and we doesn't know about how they are communicated. If you Mimos apitica, if you touch that close of the leaves because of hormone regulations. The plants you can see the distance they are talking, the trees are talk. If you know anyone listening, so they're changing environmental and changing for the honeybees and the flowering and the conditions. And this all is pollination, everything is related with the environment impact. So this is environmental impact, different variation of the flower structure, they are modified like a xerophytes, calotrophes, and these are xerophytes from this calotrophes structure. Some plants are adapted for the environmental. So there is a gene is involved. At the backside, what are the genes? They are involved in themselves, the adapting of the leaves and the structures. The insect itself, they are adapted. What is vanish? The problem of housing, the misidentification of these plants and so the find their survival, different plants, different ratio, different climatic conditions, the survival of the organism different and the plants. So this all is not a flower, this all is insect. So you have to mistake for identifications. There is this adaptation of the environmental impact. So you have to see this is a needle, it is not a needle, it is all these insects. They are identity and the hide. So in this area environment, the defense themselves they are creating for the layer, the defense of the plants and the animal cells. This is they show the seed germination of the beans. And different natural conditions, environmental, they are in fact absorption, the seed, water, if a little bit of water they add, they are absorbed. After absorption and imbibition, first their imbibition. After imbibition, their absorption, the seed coat is here and protein, the amylase is involved in enzyme amylase. That amylase is converted to the starch hydrolyzed. After starch hydrolyzed, there are proteins in the seeds. Those proteins and mad for the leaves and the root, everything. And you have to see the flight of the the wonders of the flight. This is naturally there environmentally. This is requirement conditions, so attaching and binding and maturation of the environment. You have to see that the energy of the dance of the autotrophy. And the food chains. These are different food chain from ocean to aerial, different tracing evolutions because of climatic variation and environmental. So many things are there involved for regulating that. And the speciation. So different genotype forming and environmental adaptation, changing their species. And the so many fishes, protozoa 2, amoeba, protozoa, parasalan recta, and echinodermata, mollusca, annelid arthropoda, and after prochordata, eukaryote, and cephalochordata, and mammals, and rectifiable the mammals. It's coming mammals. So all the deviation from single cells. So those all alteration of the species level, that modification of the genome level. Okay. So this species is because of environmental impact, and these are water environment. So you have to see the monkey to gorilla and man. So different ecological conditions, in fact, they are changing for alteration of the genome level and physiology and phenotype also. So you have to see that different origin of the shape is to and human holding. So this different environment, different condition, different farmers. So that different gene you have to alter and the spacing and the gene knockout and a different color you have to put the separate food. So if you example for corn, that they were wearing yellow and black and green and white and a different color of you have to put it through the biotechnology. So alteration of the genes like the expression gene is knockout or enhanced and the modified genotype and the phenotype of the corn. So different colors they have to use for attacking. So the regeneration technology in plant we have to produce much more plant and developing. You have to see that. So same human being, different environmental condition, different atmosphere, that different phase they are modified. So you got to see Asians, Russians, Chinese, Mongolians, and Canadians, Americans. You have to see the phase, different phase. So the ecological environmental factor directly or indirectly affecting for the everything. Okay, we have to study in future how the human living of the environmental condition, age factor, environmental conditions, everything. So you identify the youth for the fox. They have the range that adult gene involve aging. The aging very important criteria now. So in now recent one news that published from America, so that hug money that invest, so that scientists to improve the human life and lives is increase the 180 years. So they are going on the research. So what that it's basically on the DNA mutation arrest or the DNA replication enzyme alteration or histone modification. So if you increase the age, you have to improve the histone deacetylation and the histone modification. So the histone is very, very important now. Okay, the future has several slides available, how they are modified the histone and how they are several muscular dystrophy symptom, bone marrow thing, and the skin loss, everything you have to manage for alteration of the gene and the histone and association and the DNA enzymes for the chromosome, several enzymes, they are a decondensation of the chromosomes, they are involved at the degraded of the end of the chromosome. So this all is a problem for aging of the human being. After a year, their skin is completely damaged and shrinked. All the skin are shrinked. So this all is now they are identified the fox gene that involved take the and the genes to use the human being to improve. So many research are going on now. So Charles Darwin, that evolution was the adaptation is very, very important, and environmental factors are important to changing for and the genotype and express the genotype. And we have to think about how in future 21st century we have to alter and we have to study of the molecular mechanism of the gene, how they are expressed, what are the impact, everything you have to study that. So this is the structure of DNA. So you have to alter. And adenine go on in cytosine time. The base pair of guanine, base pair, and cyto and base pairs, and you have to think about changing the alteration of the gene structure to improve that. So you need to identify the gene, which gene you have to modify, how they are environmental, what the gene is available in environmental, how to modify it. It's like recently the spreading of the coronavirus is how they are newly comes in the earth. Is after 100 years completely suppressed, and after 100 years is remain it will come. So many diseases like that. So what is impact? So the environmental is a mutation, continuous mutation, that vigorous of the pathogens they are attacking the human. So this every 100 years, every 50 years, and the spreading of the in the world through that. And this all impact. So I have to select a particular things. So how the impact of the changing the phenotype and the genotype changing, it's express the phenotype. The genotype alter. So because of histones. Very main reason for the histone alteration, the very important for biochemical genetic approach and genetic control of sensitivity of the environment. We have to control of sensitivity to environmental factor. Like under the phenotype individual different, the sensitivity may be genetic identifications. So the phenotype for individual different sensitivity to identify the genotype alterations. We have to alternate approach to environmental and modulation of path from the genotype to phenotype. So what is environment? The microenvironment, the environment around us and the living things and non-living things. Like that microenvironment and the macroenvironment and independent. You have to see the vital micro environment, the macroenvironment unmeasured, random, and this twin model. Macroenvironment is measured or fixed. SCS, life event, exposed smoke, everything is controlled. And they are modified genotype in correlation with the genotype and individual active and evoke active or relative, passive or non-passive. So we have to identify that the involved animal and plant and microorganism, all the proper genetic system to variations and that environmental genetic environment and genetic correlation across the environment we have to find out. And what is phylogenic gene affecting for response to and widely distributed across the genome and in the animal to animal, we have to identify it for random sequence analysis. So what are the gene, what are the area, so how they are modified, and the phenotype and genotype we have to confirm. Some genes affecting response to specific environmental, like a feature sodium, calcium, rain, diet, and the separations, and the sort of minerals. This minerals is very important for some minerals, so the affecting for gene expressions. Some gene affecting response to overall quality of the environment, like environmental stress and adaptive own genetic architecture, like a genetic variability, resistant and reliance and sanitation, seeking and harm avoidance. So these are all factors that involved that. So these all impact and they are modified or modification of genome level. So what is genomics? What is genome and genomics? The genomics is first and included in the 1920s and a simple by combination gene to find out in the syllabus in the chromosomes. So the chromosomes is present in nuclear gene, okay, and the plasmid is a non-nuclear gene. Some organism genomes is defined as a haploid genetic compound. So it is cells. Genetic content organelle of the cell is not considered as a path of nucleic genome. Some diploid organism also sequence variation exists between the two copies of chromosome present in the same cells. The genome is ultimate source of information about the organism. So several organisms we collect the information and stored to a database system, several database systems. And the genes are unit of the genetic informations to present the DNA and the chromosome and chromatin. So all the chromosome and chromatin coil with the histones. So these histones and the genome DNA containing as organism cells, which including the chromosomes and the DNA, the mitochondria, and the chloroplasts in the plant genomes. So this is a structure of prokaryotic genome, the genome level E. coli, and 4 in 10 power 6 base pairs, 90% DNA is a coding genes, and a lake of membrane bound nucleus, circular DNA, a supercoiled structure of the prokaryotics. The histone not present in the prokaryotic. This is very, very important. And these all the linear circular molecules and supercoiled molecules, the DNA, the histone is absent in the prokaryotic cell, is present in the eukaryotic cells. The prokaryotic genomes generally contain the large circular pieces of the DNA. It's called a chromatin or chromosomes. Some bacteria have linear chromosomes. Some bacteria has circular chromosomes. Those circular chromosomes, DNA structure called is a plasmid. The plasmid is uncoiled structure, is a autonomous replicating agency. They have the bone is open OR region and the replicating bone. This is and swapped between the neighboring across the bacterial genomes. This is contained the eukaryotic genome and more yeast cell 1.3.35 into 10 power 7 base pairs. So almost all the fraction of the DNA total and the total DNA encoding proteins and may respond for non-coding sequence. So this DNA contains coding sequence and non-coding sequence. Most of the eukaryotic genome is non-coding sequence. If you 195% is a non-coding sequence, only 5% only coding sequence. Those coding sequence for 5% only regulate for all cellular activity, where replications and metabolism and catabolism, everything they are so all the chromosomes are containing membrane-bound nucleus, and the DNA is divided between the 2 or more chromosomes. A difference, and a set of 5 histones that packed in the gene expression it's regulation. So the packed gene expression regulation is a histone, and based on the chromosome number, the cytogenetics to identify the karyotype. Each species characterizes number of chromosomes, like a 40, rat 42, rabbit 44, human 46, chicken 78. 88 bacteria, 1 chromosome for fruit fly, 8 garden bee, 14 chromosomes, yeast 6 number of the chromosomes. So this is a number of chromosomes available that is called the karyotype. And this already you know about each one chromosome gene situated linear order. Each gene is of locus. Gene is where is available is locus. So we locus, homolocus, and heterolocus, all the region they are with different tandem repeats and so many repeated sequence and non-repeated sequence available in the locus region. Example is the bacterial chromosome range 0.6 to 10 base pair, and megabase pair size archaeologicals from 0.5 to 5.8 megaspaced per each eukaryotic chromosome range 2.9 to 4,000 megabase per each chromosomes, eukaryotic chromosome. So the plant genome size is different, different organisms, different that. With 10 MP for Astrotus, 54 MP Carlianampo, 64 MP Generis Ariosa, 125 MP for Arabis thaliana. It's a list. Arabis is a very important plant for molecular biology. So studying for the very 45 days or 60 days, 90 days complete all the life cycle. Then so only 125 megas per you got analysis successfully. And 500 megabase per Warisa Sativa, and 50,000, 5,000 MP and Hordium, 17,000 MP Triticum, 84,000 megabase per trillion large, and 140,000 MP for Paris. So the most angiosperm is a different, almost 33,000 times. And different factors, they are controlled for the environment. So this is the order of the gene organism, different order, their arrangement for the bacterial and just eukaryotic, monocot and dicot is a variation of the genome size. So C-value paradox. What is C-value paradox? The total number of the sequence that is called C-value paradox. So the genome size is the base pair 0.9 into 10 power of 9 into DNA content. That is called picogram. So the DNA content is waiting for picogram. Genome size is a base pair, counting the base pair. So 1 picogram equal to and 9,000 base pair and 910 base pairs. And it's called 1 mega base. That's 1 mega base pair. 1 base pair equal to 600 Dalton. So this all is conversion of the value of the paradox of C value paradox. The sequencing genome, duplication chromosome region, medium repeated sequence, non-repeated sequence, highly repeated sequence, tandem repeated sequence, everything is controlled for the and some genes inside, some genes outside. The outside means the environmental factor. We have to, what are the gene is control the outside environmental factor. See that one by one in the next coming. So I selected only one how they are influence the environmental factor alteration of the histone. The histone very, very important for the coil around the DNA molecule in specific manner. So this histone packed 2 micron of the DNA is nuclear with the diameter 5 to 10 micrometer. So you have to see the different size. First the threaden of the chromatin, chromosomes, and after this model of the chromosome, each one and after they are with the coil around histones. So the histone environmental factor impact and the histone how they are modified, how the mutation, how the genome modified. This previously you see this so many modification, so many features development, everything is a histone modification. So histone and the nucleosomes. So what is chromatin structure and the remain of the chromatin nucleoprotein complex? So this condensation of the organization DNA, the protein and the DNA. And without the chromosomes is contained for the protein and the DNA. The protein is a histone, the DNA is around that. The protein DNA, the ratio is a 2.1 and the histone DNA 1:1. So one portion of histone, one portion of DNA to coil with them. So this is called and histomer. This is called histone and histomer. Several level of condensation. The 300 nanofiber formation, the histone tile, and mediated nucleosomes, and these tiles and mediated association with individual fiber and produce for tertiary structure of the chromosome fibers. Okay, you have to see that each long and chromatin fiber linker DNA. So that 30 nanometer fiber with a linker histone H1. The H1 is a linker DNA. The short range after the nucleosomes, the 1.2.7 and core, the nuclear core, that is called nucleosome. With around coiled with the histone and the DNA, it is called nucleosome. So this core of the histone is tail is a domain. It is called domain. This chromatin is basically two types. It's a heterochromatin and a euchromatin. And what is heterochromatin? Heterochromatin is a transcriptionally silent. It is inactive. And the euchromatin transcriptionally activated. So highly condensed region, highly coding region. So most of the chromatin is a euchromatin region. If you mutated, the gene affected, they're causing for several diseases like a cancer, metabolic disorder, disease. Those are all genes is available only euchromatin region. So the euchromatin region is transcriptionally active. So this nucleosome is repeat unit of the chromatin. The repeat unit chromatin repeat length 180 base pair to 200 base pair the length. And these two enzymes is very, very important. Nucleosome digest. One is and oligonucleosomes. Another one is a core particle like chromosomes. And the nucleus activity chromatosomes is 116 base pair DNA plus aptamer plus H1. Like H1 and the protein H1A, H1, H2A, H2B, and H2C. These are all 4 core model. So the even nucleus, this core particle 146 base pair DNA is formation for aptamer. So aptamer is a blend like blend and around warp around the warped DNA is formed 1.7 turn of the left hand helix with the nucleosome core particles. So you have to see this nucleosome repeat and chromatins. So this core particle with the and this core is made of the beta chain and alba and a beta core for the C H2B core. Another core is H2A is opposite and H1 and H2B. So this core was with arranged of the molecule, the coil with the DNA. With the coil with the DNA is a 1.1. It's a K5 region, K8, K12, and 16. So many N at nitrogenous group is available that are attaching for so many molecules. So this is a complex structure of chromatin, nucleosomes and chromatin structures. So this was the H3 H4 tetramer and H2A and H2B dimer. The octocore, like a octocore, this is 4 type of the protein that is called octocore. It is called nucleosomes. Those octosomes and octocore is a H3 and H2 tetramer. This is a helical structure of that. And H2 H2 dimer. So those dimer will linked with the histone of the linker DNA, which is a H1, is a linker DNA. The dissociation N-terminal and C-terminal and nucleosomes, and you have to see minor grow and major grow. There are octomers, H2A and H2B and H3 and H4. This is called octomer. This assembled H3, H4 tetramer plus H2AA and H2A and H2B is a dimer. So this is structure of the, this is, you have to see that, and NKK, like this is a sequence order of linker DNA. The nucleosome 3D structure and the crystal structure of nucleosome particles and 2.8 angstrom resolutions, 146 resolution, best per DNA warped around the histone is octomer, and this outside position of the histone tiles, they have the another attached to this. This is called histone tile. This DNA is warped around is 1.5 turn of the left-hand helix and a supercoil model with the nucleosome core particle. This is then the Looper et al. 1997 and described for the and bioinformatics software. So this nucleosome 3D structure to 11 nanometer core is rotated like that. So if you nucleosome turning during the chromosome separation, like that turn, exit, enter region, this is center region, that is called the exit region. So this what histone and linked histone structure is H1 linker DNA zigzag and manner like that B solenoid structure. So this is a variation for solenoid structure. Zigzag is a different, solenoid is a different. So the association with the linker DNA between the nucleosome H1, this is zigzag. The binder DNA entry and exit, simulation and binding the 30 nanometer and nanometer fiber core, and H1 bind and weaker to acetyl and a nucleosome. This is structure of the H1 linker histones. So you have to see this H1 between the and the core particle, they are linked with the H1 proteins. The chromatin general repressor, so the transcription. So this is a regulate for the so many and the logic gene regulation and prokaryotic versus eukaryotic. The prokaryotic gene regulation with 3 elements: promoter, operator, positive control element. The promoter recognizes RNA polymerase, the operator recognizes repressor proteins, positive control element recognizes for the activator. So the prokaryotic genes, if you functioning, requirement 3 type of RNA polymerase, repressor, and activator. That is called ground state. This transcriptional ground state usefully concept that and regular sequence and prokaryotic and ground to active site that binding. So the binding and non-binding, that binding is active and non-binding is inactive. So this around regulating for the eukaryotic ground state and restrictive, the stronger core octomer and the promoter is essentially inactivate the eukaryotic cell. Essentially all the eukaryotic genes required for the activator. So this is due to the chromatin template and they are regulating for the gene expression. If chromatin template is a mismatch or failed, the gene expression level is a default. The two mechanisms, gene activation and inactivation. The activation anti-repression of the inhibitor chromatin and is structurally indirectly across in the requirement of polymerase second mechanism. The polymerase second is binding of the and active sites. Like that step 1, you have to see the ACE and binding protein that actives transcription start and active site and BA site. So the transcription factor require, histone nucleosome is required, remodeling factor required. If you chromosome structure which indirectly increase for requirement of polymerase activities. That is main. If chromatin acetylation, if any problem, this polymerase mechanism will stop. The gene expression level is down, and this effect involved in the phenotype 2 and genotype 2 changing the phenotype. The activation requirement, that activator component of the polymerase 2 machineries and both large targeted both TF protein, the transcription factor second and the holoenzyme. This is activating site. So this activations requirement RNA polymerase is presence and binding with the activation activator site in the active course. So the chromatin maintained for the transcriptionally and restricted ground state. Almost the chromatin general inhibitor protein access of DNA, but it's interesting different and the TPR that is called unable to bind the nucleosome proteins. So many activator binding for the nucleosome DNAs and the remodeling and activating for the chromatin structures of RNA polymerase and the DNA polymerase second mechanism, core proto and core promoters and many activator bind to the target site that is called DF secondary and DPR proteins. DPR protein that is called activator. The core particle is directly binding with the aptamer in the activator. So this, if you de-escalation of the aptamer, the expression level is a fault and the genetic alter and creating cancer. So many diseases because of the gene expression level alter the genomes. To changing the chromosome to allow the transcriptions, how they are allowed. So the changing chromatin template helps the transcription factor. Is this all the transcription factor? AF factor, NURF factor, sensitive factor, RS factor, HTC factor, HAT factor. So this all HAT factor is very very important for this and transcription transferrin factors and the SPT adult, that is acetylferrin factors, and HSCO enzyme at histone acetylation transfer, that is called histone acetylation, histone and acetyl transferase factor. Those opening chromosome to directly nucleosome mobilization of the ATP dependent process, that is adenine kinase process. Another position of the nucleosome create the promote of the different requirement of the remodeling of the structure. So that automatically the remodeling of this all is a aptamer histone modification because of HAT and HAM. The HAT is called and histone acetyl transferase and histone methyl acetyl transferase activity. The basic of histone code, the hypothesis changes. Histone acetylation. The main reason that most of the mutation occur in the chromosomes and DNA because of histone acetylation. You have to prevent the histone acetylation and prevent it's a minimize of the mutation level of the genomes. What is acetylation? Core histone. So N-terminal tile and reversible acetyl lysine and particularly H3 plus H4. You have to see this and the positive ions, and it's attractive for negative ions. So this while globular core involve the histone packing DNA content that N-terminal tile. Those N-terminal tile have the so many plus like positive charges molecule. You got to see N-acetylserine. This is structure of N-acetylserine, CH and NH3 COO group, and CH2 and NCO and CH3 group. So this HAT is a reversible reaction in the HAT and the HTS. And what is HAT? The HAT is a histone acyl transferase, and another is a reversible reaction like that lysine. So acetylserine to lysine, lysine to acetylserine, that modification only, that acetylation, acetyl group changes, their core is, they are mutated, and the mutation occur in the chromosome, the replace of the particular base pair level. So this is a acetylation. Another is a consequent acetylation of the amino group, like lysine, lead to the reduced positive charge. This is positive charge, and believed that weaken the DNA interaction with the tile region only. So if you change the conformation of the nucleosome structure and disabilities of the intercalations of the contact, so the removal of the histone tile result in nucleosomes, the arrays cannot condense it and pass 10 nanometer fiber. So that is the chromosome damage because of the acetylation core. So many changes interaction with the regulator protein in the regions. So you have to see the changing of the change to change. This is structure of that. This is a nucleosomes nuclear core is a tiles histone coenzymes hat. The histone lysine CH2, 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 NH3 group that is called histone, lysine histone, and coenzymes SCO and CH3. So this both are is regulated hat molecule. The hat is histone acetyl transferase and with histone acetyl coenzyme and modified the histone lysine. So those histone lysine they are altered at that changing of the change in the chromosome types. So this is you have to see the plus molecule and O group. So histone acetyl and transcriptive. Acetyl histone and tile enriched transcription chromosome. So that becomes acetyl acetylation to make DNA core and acetylable TF finding factor, transcription finding factor, roll up and potentiation. The histone acetylation with the linked transcription regions. So this is gene regulation, how they modify histone to functionalize. These are modern enzymes, specificity. This is octave core. This is a core particle binding with the protein that, and after starting the functions. This enzyme of the hat, how they are enzyme involved. Histone acetate transferase. This is the three-dimensional structure of histone and protein structure. These are enzymes. Most of the enzymes are protein, but all the enzymes not yet proteins. And YGCN3. This is structure of the helical structures and tubular structures. The helical structure like that. Hat molecule. N-acetylations. Those involved is a histone acetyl transferase. The catalyze transfer to acetyl group to acetyl GoA group into the histone ties. So this is a major reaction occur in the histone acetylation. The histone methylation, methyl group transfer, methylations, histone acetyl group transferation is called acetyl transferase. So this catalyzation process and transfer to acetyl group from the acetyl GoA into the histone ties. So these two major group acetyl GoA and A type and B type hat. So this hat molecule is histone acetyl transferase enzyme, and this catalyzed trogene is related to acetylation. Trogenes, most of the trogenes available in the cockroach development segment of development gene. It is called trogenes. And the trogenes, where most of the development biology they are studying for histone, lysine, N-methyl transferase, and thyrox. This is the expansion of the trogenes. And histone, lysine, N-methylase transferase transfer is a trithorax is involved of the segment development of the Drosophila melanogaster. Drosophila also that another one lower group. So these two main group is hat enzyme, coenzyme, cytoplasmic catalysis acetyl linked transport synthesis of the histone from cytoplasm of the nucleus. The histone deacetylase that is called HDACs. So that histone acetyl transferase. If you transfer the acetyl group, transferase required and remove the acetyl group requirement enzyme, it is called histone deacetylase. If you mutation, automatically they are binding the sequence and methylated group to remove it, the methylation and acetyl group, and like that, deacetylase the find out remove that. This is this core proteins. So this one three type of and HTA like that. So this one is and H hat and had histone and acetyl transferase. They have this enzyme is a 3-4 core, 1, 2, 3, 4 core like that. H hat 1, YGN35 and HGNC5L and HBCFC1. So this is a different octocore like that, 832 nanometer and base pair, 800 base pair and 400 amino acid base pair and 370 amino acid. So this 4 core is together, that is called the genate family genes, that is hat genes. So most of the hat genes is involved for the acetylation, that is called octocore acetylation process involved for the hat genes. So you have to see 2 mega-Dalton complex, the 4 subunit TAF, and those interaction TAFI and the TAF and binding protein that act acetic activator, and the human genes GCN5 found in similar complex stage is available stage. What is stage? And stage is SAGA, that is SPT, ADA, and GPG island, and acetic transferase, that is expansion of stage. So the HAT enzyme found in the large complex, you have to see this, all the HAT enzyme found, core protein genes is successful. So many binding core available is involved for the. So if you study the molecular biology and how they are genomic sequence to identify only HAT enzymes, what are the work in the different features and how their modification structure of the genome level, how the phenotype expression, genotype expression variation because of HAT enzymes. So you have to study in future research. So you got this, you have to see that PSF and the stage and the tip and the tick. So you have to see the HTP 500, so many proteins. Those so many core protein that HSP 70 and HSP 3 and HAT BR2 is present in human. This all genes present in human. This all present in human. So that acetyl very HAT enzymes found a large complex for the all animals and plants. So the activator, that is called activator content trap genes, activators. Structure of HAT enzymes. The transferase and the histones acetyl transferase structure. The histone acetyl transfer domain. That domain is and the central conservation core unit important acetyl CoA factor binding that. So if you clearly use for substrate recognition to bind the directly and over cofactor packet and the expression of the genes. So this HAT structure is a peptide binding packet. So peptide core. So peptide means more, and one peptide means more amino acid that together joining the formation of the peptide. So this peptide is a H3 peptide bound and a random oil structure, everything. So this extended packet contain, and this is a phosphorylation, serine and H3 peptide, and generated additional attractions. So those explains histone modification can influence of creation for another modification. So very, very important. Histone, if you modify it, is influence for so many mechanism modification. Phosphorylation, serine, and so this is enhance of the lysine 14 amino acid. So involved acylation of the together promote of the transcription factor. So this is a HAT involved on the gene regulation. So if a large number of HAT complexes found that if you that inhibitor, Tata box, and the TIF transfer factor, and the TIF 250, and their binding factor is SRC1 and P30, 300 CPS, and ACTR factor, enhancer, the operator. So several cofactors have the HAT activities. The HAT, if you activate so many cofactor required, HTFE 250 and the TAP 230, TAFE 130, and B300 CPP, and the ACTV are activated, and BSCAP, CF, and the gene 5. So these all involve the HAT complex formation. So those HAT complex gene specific expression involved. So this is the mice and the P300, so many knockouts, and GCN5 regions, the knockout gene is expression, the distant development of the defect genes, everything you have to identify the HAT complex involved. So the development biology, you have to study how to modification of the and the development of organism and the small to mature level. So you have to study histone modification, how they are histone modification. So the core particle H3, H3A, H4 is together joined H3, H2A, and H2B. So this involved required for epiquinone. The epiquinone is a catalytic protein, like a sister molecule that protein. The protein is binding. If you binding or DNA sequence, that sequence is a binding is a catalytic epiquinone mediate sequence removal. So this epiquinone three major method involved. One is methylation, another one phosphorylation, another one acetylation. So three factors involved for histone modification. Those histone which lysine modified, whether histone or nucleosome or substrate modified, or free genes, acetylase free histone, they are modified, and SAG complex modifies the nucleosome. So this all involved in the SAG and the nuclease gene 5 involved in the histone modification, like a 3. So the blue color is a methylation, and the yellow color is a phosphorylation, red color is acetylation. So far we are finding protein, modified enzymes, and acetone score, and whether functions, if you binding with the active core is a function. The binding protein they are finding with the active course is a function. If you're not binding, no functions. Like that, the requirement of the missing link. So the HAT with the enzymes and like a histone acetyl transferase were found a long time out considerable interaction trogens, and this trogens development biology is in Drosophila because of the assault of the active global, that Tata box, the transcription fox that is called Tata. The cat activity co-activator complex and promote the and the transcription factor interaction, the gene inhibit and activate the gene expression level. So cat complex, the Tata box binding that, and the cat complex and general transcription factor that consists of the protein elongation and other cat genes. So this is a factor involved, and the cat is very, very important for the cat and the histone acetyl transferase enzyme, and the co-complex is the binding for the Tata box, and so this is a core protein. So the global acetylation and the target. The worst target acetyl de-acetylation. So the bulk acetylation level is surprising high. The average 13 to 30 tile lysine residues histone octamer acetylated. If acetylated, is damage of the acetyl group and remove it acetylate. And another de-acetylase enzymes involved for the acetylations. This stays the stable level acetylation maintained by the opposing action, like that positive group and a negative group complex. They are forming complex, so that is called the HAT and the HDase complex. So those targeting HAT and the CAT complex, the chromatin region, they create that in specific pattern of the hyper and the hypoacetyl background and the global acetylation of the correlate with the transcription activation and repression and respectively in the targeted and group of the protein conformation changes in the octocore, like that acetylation and de-acetylation. You got to see that, and use the Tata box and all this ADA group is the SH group. So the SH group, so many active factor there, active group there, core particle, that is called core complex, CAT and the co-activator complex. The SACH complex is SPT, SP220, SP18, SP13, SPX, and ADP, ADA, and ADGGN. All it is called SACH complex. The SACH complex SPT and add GNC acetyl transferase group, that is called SH group, ADA group, and CAT acetylation co-activate binding. That is co-activated with binding that and activation, that's called aspartase complex and SAGE complex, formation of the SAGE complex. So two activated, one is CAT local acetylation co-activated bridge method. So two type that CAT local acetylation of the method to this removing for the acetyl group in the. So the binding protein with the bind that function as binding protein, the function is present and the gene is expressed. Bromine binding bond will acetyl histone. The bromodiamine recognizes acetyl lysine histones. 100 amino acid is motif. The motif means what? The motif is a small amino acid is a binding protein, and a binding protein that is called motif. The structure of motif is the chromatin as a protein including CAT, like the structures. The four helical, the motif, the bundles of the hydrophobic packet and interact with the acetyl lysase. So it's a promo H4 complex. This is and the beta chain model. So the promo and the CAT logic in order requirement of the promodine complex, pro-complex synthesis, the binding nucleosome core particle, the nuclear core particle, the express of the and the CAT loci in the development of the biology. The requirement of the further association modifications and the targeted SAGE acetyl histone, H3 and the density of the promoter, targeted acetyl SAGE. You got to see that and how their core particles bind with the enhancer, Tata box, and the tile is a tiles and the H3 tiles. And the targeted acetylation SAGE and binding with that targeted and SNF chromosomes, single nucleotide phase demodification complex, they are binding and they are activated. So this requirement proto and domains gene 5 and SUB2 and representatively which is acetyl lysine, that is called acetyl lysine enzyme, acetyl lysine group. So due to the bromodomain, other protein acetylation might stability of the interaction of several complex their formation. So this is associated with the modification of the histone like that. So this binding protein, this function function is binding that several CAT groups and in large and a co-activator complex, and yes, the specificity, correct specificity, specifically find out. And this complex pattern they are finding all the four group promodomain binding complex bind with the and binding proteins. So that is TRA activation. If a TRA activation gene is a Drosophila genes, and bottom of the complex, the development biology, and you have to see that what are the factor requirement of the and binding protein is a bromo domain proteins and TRA activating genes for the modeling remodeling structure of the HAT is active. That is had enzymes and the divested acylation enzymes, that is called modulation enzymes. Histone modifications. The histone modifications is two types. One is phosphorylation, another one methylation. The phosphorylation means the stereomitosis and associate and EFG H3 stereo and mito associate and EFG stimulate RSK modified protein. So this phosphorylation is find out S10 region. The S10 region and H3 protein core, this phospho and S10 may be enhanced acetylation surrounding of the residue. That is called a S10 residues. This S10 phosphorylations. So this histone modification involved and with mediated epiquinone mediated and ATP ribosylation, adenine triphosphate ribosylation, that is called ribosylation. So this all mechanism, the phosphorylation and region specifically S10 and is involved in the histone modifications. The histones modifications among the most conserved region only. This protein now this conserved region may be evolution this organism for different changes genomic level and the most variable in the post translational modifications and the H3, H4 and H2, H3. So this all is histone modification, methylation, phosphorylation, acetylation with the epiquinone mediated. Histone methylations. What histone methylation? The histone is an octocore molecule in the lysine N monomethylation modified HMT enzymes, and after is a N-methylserine converted and N-methylserine lysine. So these are finals. And we got to see that histone methylation, both K and ORH and R can methylated than one methyl group. Histone methylation relatively stable modification with a low transfer rate, and no histone and dimethylase are found in the lysines. And ideal epic mark of the long-term maintenance of the chromatin stress, and this because of histone methylation. There's multiple variant lysine, arginine involved. The methylase residues and presence both in and euchromatin and heterochromatin is found. So the methylation, one of the very, very important is the mutations. If you methyl and methyl ligase, the methylation group, if you identify the methylation, if available that DNA sequence is modified or not, in easily confirmation of the sequencing identifications. So the substrate histone arginine lysine methylation. So the argine lysine, the arginine with the acetyl and the prime type of this convert to the monomethyl arginine. So the changing for acetyl CH3 and arginine methyl group CH3 and divide into the asymmetric group and the symmetric group. So both is different with the symmetric dimethyl arginine, asymmetric dimethyl arginine. Lysines, this amino acid modified lysines and monomethylate lysine and dimethylate lysines. Monomethylate, dimethylate, trimethylate lysines. Those increase in the besides of the lysines. So these factors, we got to detailed studies of the methylations are available in the histone and histone because all basically two region only methylated. One is arginine methylation, another lysine methylation. And another one, the enzyme histone lysine methylase transferase, that is called HKMTS. The HKMTS means what? That is enzymes involved histone lysine methylation. Lysine, all the lysines group methyl transferase, the methyl group transfer. That is enzyme, it is called methyl transferase enzyme. The first KHMTA is identified 2000 only. The specific methylase L19 and the region, that is 9, L lysine 9 and region, the modified histone H3. So this larger lattice and larger level HTM identified as several organisms like site-specific and Saccharomyces cerevisiae and S-Foamy, Saccharomyces melanogaster, Drosophila melanogaster, mammals. In the several mammals, they are identified the gene HKMT activity for set domain. That is called set domain. 5 amylase, 5 lysines with H3, K4, K9, K27, K36, K97. So all the core is a methylated specific. That. Attend பண்ணுங்க. attend பண்ணுங்க. அதுதான். So most HKMT, that is enzymes, conserved set domains. The region is set domain, that is called set enzymes. And this structure of DIM5 homology find by methyl lysines and cofactor with the zinc ion. So most of the cofactor with the zinc finger. So this core is present the zinc finger. It is called zinc finger. And this is coiled and helix and coil structure. So this is set complex. This set domain is methyl lysine 9 or H3 complex formed. The blue color H3 is a blue color and the cofactor is a purple color and the zinc finger is a pink color. This is a zinc finger complex. So what is function that long epigenic maintain of the epigenics? The epigenics maintain histone only. The epigenics. Heritable changes the gene expression and that do not alter the DNA sequence. And HTM is involved in a complex process that control accepts the short and long-term transcriptional regulation of the genes. So this function is very, very important for the genome variation and species variation and the formation of the evolution. Everything is epigenic. This heritage changing gene expression, alteration DNA sequence continuously in the form where at HMT complex process molecules. So you have to see the activation repressions, step of methylation, and you got this is a sequence order and the histone methylation groups, and a K3, K9 methylation correlation, heterochromatin formations, and methylation of the K4 in the H3 correlation, triactivating gene. So one is a repressor, one is activated trogene, another one repressor. If you bind one group with 4th region methylation and activate for the trogene, another methylation 9 region G group, there repression of the trogenes. So trogene is most of the development biology of the melanogaster and the formation of the segment. And this trogene activator, so that complete detailed study about associated with the heart complex of development biology. For what are the function of histone is methylations, and most of the repression H3, H4, and the number of report silence that is involved of this gene silence activity. So gene silence activity involved for the tomato ripening. If you RNA antisense RNA technology, they are involved for the RNA complex. The gene silencing or zig parallel, that antiviral. If you anti-DNA active, that is RNA and blocking for another one RNA, doubling RNA. That is called silence or knockout of genes. So this region, H3, K3 methylation region, they are used. Another, the heterochromatin formulation link H3, H9 methylation, H1B binding protein, highly condensed and centric chromosomes and the methylation group. So this is important for X chromosome inactivation process and histone and histone methylations, and very, very important for X chromosome inactivation process. Activation H3, H4, and methylation generally associated with the DRA activation chromosomes. Dimethyl H3, H4 appear in global epigenic and euchromatin regions, and DRA methylation H3, H4 for K4 correlation activation for the transcription regions. So this all is a spreading heterochromatin region. They are find out euchromatin regions and preventing spreading of the heterochromatin region. So this activation repression is very, very important for function of histone methylations. The role of histone methylation and the DRA elongation factors. So this is a cofactor with serine, sesvicis, NUDA, and the toxins. 4 protein activate of the serine and activation methylation 2 level. The chromosome level, heterochromatin level, gene level, and the elongation more level, gene level. So This recognition to find out the chromo H3 complex models. So this all factor involved chromatin bind histones and methylations specific manner with high activity, lower activity of the gene expression. So this all methylation recognize specific chromodomains. This all is a transcription silence of the HTTP level, and this all structure of HTTP molecules and the prodam catalase that these are all functions and H3 histones methylations, and where removing and histone methylations considerable irreversible epigenetic region, histone demethylation regions, everything. So these are all summary, you want to see summary, and binding protein with the functions. So yes, complex draw activation repressions. So this is model. So many, and I skipped that several very. That's what that I go to mental genes more than just DNA modality. So the mental and gene expressions, and they are find out several research. So the how they are the sequence of the biologically modifications, like biological event, local structure of genes, binding site of the proteins, epigenetic mark. These are all activation, inactivation part camped, HTM, kinase phosphase. This all group, this histone modification changing short-term local structure of the chromosome is the binding site of the protein. That histone modification epigenetic mark, euchromatin and heterochromatin. This all changes involve the biological event. That is a mental, and the gene is passing for generation to generation in the maintain for epigenetic region, euchromatin and heterochromatin. All the biological event only depend on the three enzymes, HAMT, HMT, and kinase phosphate. So nuclear disruption, what that functions altered. What you see this structural disruption, remodeling that TF gene, remodeling of the complex, and this unwarped. If you the warped and unwarped, that is involved for the DF factor, reducing DNA supercoil nucleosome. The nucleosome disrupted by the SWIFT and SNP factor of the genes. Nuclear disturbance of the SWIFT NF. So this intermediate complex to binding that, if you remove it, that you have to see that removing, and the binding for the DF factor that activated. Modeling and remodeling, silence and transfer involved in the histone modification. Without histone modification, that two factor there changes. One is cis factor, another one is a trans factor. The cis is siding, and the spooling and the twisting, that is our transfer, is a transfer gene. The remodeling nucleosome can happen by the local disturbance along with the stem structures. So we got this two maintain mechanism of the histone proteins. One is sliding, and the conformational chromatin, octamer, DNA path, and phosphorus. So these conformational changes intermediate like that, changing for octamer region of the chromosomes. So this you got the two type of mechanism. One is a twist defense mechanism, another pulse defense mechanism. Two mechanisms involve the alternation loci because of the this twist of the local alteration around the nucleosome level. So what this two model, they are proposed two model. One is a Swiss synapse properly model, genome analysis is 5% all genes require a SWIFT and SNP model they are find out in that. So these interactions help the activate of the gene with the octamer like that, helping of the sequence step, genes, the octamer core, the find out, and the three octocore, SWIFT, ACT, and the TPR, and the gene activations. So this implication of the order activation and the principle of gene regulation for these octamer genes and the SWIFT and binding factor and cell alias. So this is structure model and active transcription factor activations, and left that transprotein activator with associated gene expressions, and that gene activator through the activation associated with the promoter gene when transcriptionally active. And epigenetic memory, the chromatin modification activity required can remodel after and transprotein has left constitutes on the epigenetic memory. So the condensation, decondensation of the chromatin, two very, very important factor during the cell division, cell alteration, and epigenetic gene memory is very, very important factor there. So of the gene path, you have to see the ORI or gene path, how they are condensed, gene transcription factor with that modification of the DNA dependent remodule covalent modification of the promoter. And the individual promoter working at the set of order action of those complexes different from promoter to promoter. So SWB, SNF complex, CAT complex, and regulator sequence with acetylchromatin chromatin regions, accessible regions, and a specific order their arrangement condensed of the chromatins. So this is called gene species pathway. Resolution paradox and orthoparadox and required for the and the transferrin involve the chromosome alone. So with the cis element available, transferrin without disruption of the nucleosome, without disturbance. So other hidden that element require nucleosome disturbance, displacement before the DF binding. And after transferrin with multiple complex cooperation fissuring that inversions. So this all is alteration paradox and the cofactor with the activated proteins and very, very important complex transferrin binding protein. Its domain is activating for the and the chromatins and the condensation paradox and orthoparadox methods. And these slidings for conformational changes sliding under intermediate. This is intermediate of the complex. The represented by the SV and P, that is genes, genes activated, and repressor protein gene activated, and genes that gene activated. Like that, they are repression and non-repression. Nucleosome position. So this is a pair length transversion and a rotational position. So how the rotation effect of the nucleosome depend on the position. So this is the inward and the towards. This is called pi angles, pass sides. The nucleosome positions are important positions for DNA coiling. DNA nucleosome is coiled, having specific translation positioning and a position dependent particular DNA sequence partially bound to the transferrins. So those DNA varieties, flexible venerative bending octomers around that performs positions, that remodeling and the thermomesmic AT rich region, minor growth forming region, GG rich region. So very, very important factor was a transcription factor with the and the serinucleosome. So that arrangement of the nucleosome in position, it is called translational regions. So they are how they are arranged, TF factor and the PIC. That the transferrin factor, that binding with that, this is not good, it's correct. So the binding with the nucleosome with the periodic geometry and grading and create for and binding sites. So those nucleosomes are stimulus interaction required for the transcriptions. So the nucleosomes and position of the nucleosome and the determination availability site and the representation third language sites. You have to see the hormones and GR goes to binding rotationally active and GRE complex with the SNF complex and dissociation. So there is a representation. This is determination available of the site, a role of presentation, cis element with the bi-base slicing of the pi base pair, slicing representation element with the changing of the pose from the out. So this is model histone variant with the H2A histones, complete H3. This all blue color H3 and yellow, green color H4 and H2B is red color and DNA the H2A is light. So these four different color to indicate of the and nucleosome has distinct feature and metal binding site. So those metal binding site and the positive involved for activation chromatin promoter. So 60% conserved related H2A. Most of the region, conserved region percent in the conserved region is a not mutated region, conserved region. The conserved region, most of the H2A protein available. If you have other protein, they are most of that mutated region, that is not conserved region. Non-conserved region easily mutated that sequence because of the modeling, remodeling, and maintained by the histone only. This histone influence of the DNA mutation, alteration, everything under control of the histones. zDNA. So that the aDNA, bDNA, and zDNA, that conformation changes. This is threatened and the size of the zigzag manner. That conformation stabilized with the supercoiling structure, activator protein, the minor group, transition, transpiration, major effect, chromosome template. The sequence is identified like that, and the 60 base pair pro Tata box 2 is 4 base pair pro Tata box in the 85 region, 125 regions. So this model closely conformized PFD complex, NFCFC complex modulation, and zDNA complex with the nucleosome formation different. And that is in a open conformation, that is called open conformation. These mammalians like WN1, SNF, like that PFD complex coordinate the DNA binding protein induced for the DNA. So these are all open conformation, these are closed conformations of the two type of the DNA. So anything, this naturally the DNA structures like alterations and mutations involved in the directly, indirectly up to the acetylation, methylation, and involved in the October, that is histone, nucleosomes. So most of the environmental factor, genes, and directly, indirectly involved in the nuclear genes. Most of the organism nuclear gene is a mutation. That mutation continues in the world and environmental impact and so many fishes and so many animals that separate out living things, everything. So anything you have to save the nature, it will carry us. And so thank you for giving the opportunities. Thank you one and all for listening and this.
Thank you, sir.
Okay.
Thank you, sir, for your informative session.
Okay, thank you,
Forum is
madam.
open for discussion.
Any doubt or any questions?
The forum
There
is
is a
open for
question in
discussion.
chat box.
Sir, can I ask a question?
Yes, sir.
I'm Dr. Pradeep Desai from Ahmedabad.
Okay, sir. Thank you.
So my question is related to this Corona COVID-19.
Yes.
Now, how they are able to mutate so fast so that waves 1, 2, 3, 4 keeps on coming?
Sir, almost
Are
prokaryote
there,
genes.
and second question is there any DNA programmed in such a manner that they can manipulate?
It's a good question, sir. Is coronavirus basically that prokaryotic, isn't it? It's a prokaryotic. The prokaryotic gene easily possible to mutation. Very, very easily possible mutation. Any prokaryotic. If you almost mutation study, we have to choose the prokaryotic cell and a small sequence base pair. It's a very 100 and 120, 180 base pair, easily mutated. This mutated only region for spike region. Spike region. The spike easily they are holding the mechanism, the increased spike and attachment in the our bodies, our cells. Attachment, they give the more spike. That is only modification of the coronavirus spikes.
Yes, sir. These spikes are protein based, huh?
Yes, protein based. Protein based.
Thank you, sir. Thank you.
Thank you. Thank you very much, sir. Any other question from audience?
Participants, if you have any questions, please unmute yourself and you can ask. Raji?
Ma'am, shall we move on to the next session now?
Yes, ma'am.
Okay, ma'am.
Thank you, sir,
Thank you, madam.
for your excellent presentation on gene and environment. It was an elaborate session. Thank you. Hope the participants would have been benefited by this.
Okay, thank you for providing the opportunity. Thank you once again for all and listening participant and the students, scholars, and professors. I thank again. Thank you.
Thank you, sir. It was a very excellent section,
Thank you.
session, and everybody have benefited from it. Thank you.
Okay, madam. Thank you, madam.
Now, we will begin the technical session by introducing the resource person, Dr. S. Muralidharan, Senior Principal Scientist and Head Division of Ecotoxicology, Salim Ali Center for Ornithology and Natural History and Director in charge. He has 36 years of experience. His area of specialization involves ecotoxicology, environmental chemistry, risk assessment, and wetland contamination. He received Fulbright Award under the Indo-American Environment Leadership Program 2003, Special Achievement Award from Petrus and Wildlife Research Center, U.S. Zoological Survey, USA 2008. He published 30 papers in both national and international journals. He published 2 books and 16 reports and presented 62 papers in workshops. He completed 22 projects and 3 projects are ongoing and guided 8 PhDs, 4 MPhils and 65 MSc dissertations. He served as a member of the Board of Studies, member of editorial board of the journal Ecotoxicology, member of technical committee of Tamil Nadu Agriculture University and Bardiyar University, member of Indo-Cell Phone Technical Cell constituted by Kerala State Council for Science and Technology and Environment. He is also a recognized guide of Bardiyar University Coimbatore and Manipal University Karnataka for MPhils and PhD program. We are fortunate to having you, sir. The session is over to you.
Thank you, madam. I will just share my screen now. Audio only. Video on video. Hello.
Yes, sir.
Am I audible?
Yes, sir. Yes, sir.
You are able to see my screen?
Yes, sir. Your screen is visible, sir.
Are you okay?
Your video is not there, sir.
1 minute, madam. I am trying to solve it.
Yes, sir. Yes, sir.
Hello.
Yes, sir. Now it's fine, sir.
Okay, everything is set, I guess. Okay.
Yes, sir.
Okay. Good afternoon to. I think it is just 5 minutes to 12, so I would still say that we should celebrate the morning of this pleasant day today. Good morning to everyone. Thank you so much for this wonderful opportunity to speak to all of you on this occasion. When I was contacted to give a lecture on this, I was a little skeptical as to whether I'll be able to manage time because of this 31st March, which is around the corner, closing of accounts all those things was really a kind of situation which was not allowing me to immediately give commitment. Then anyway, I guess everything is now in line with our expectation. I'm really glad that I'm able to speak to all of you today. So the talk I'm going to be giving today is on unsustainable use of chemical pesticides and their ill effects in the environment and birds as a touchstone. So this being the topic, I guess many of you would feel this topic to be totally different from rest of the lectures which are organized for this workshop. Well, in Indian context, if you look at, not many people are looking at the ill effects of chemicals and then again focusing mainly on birds. So this has become a kind of a topic which has not attracted many researchers in this country. And first question anyone would ask is that as to why birds should be affected because of chemicals, pesticides which are being used by farmers to contain the pest in the agriculture crop. This said, the simple answer to that is that birds are not found only in the forest or naturally protected areas or sanctuaries. They are there everywhere, wherever there is habitat, and definitely birds would go over there. And then on the other hand, if you just look at wherever chemicals are used, soon after rains, they are going to get washed down to the nearest lake or reservoir or any wetland ecosystem where birds are inhabiting. So that way, they are going to pick up these contaminants and then they act as the best indicator. So this is in brief as to how birds are affected because of these pesticides. Before I proceed further, you might want to know as to what I'm going to be talking to you in the next 45-50 minutes or so. So when we talk about chemical pesticides and the real effects on the system, their awareness or the eye-opener scenarios which happened in the whole country would coincide with the publication of book titled The Silent Spring by Rachel Carson. So this book, in fact, awoke, or rather created the kind of awareness among people as to how these chemicals can create problem to different species of birds. So this author, Rachel Carson, is not, was not rather a scientist by profession. She was a US government employee who had little interest on birds. So she used to be observing these birds in the backyards of her house, where she had a little space with a lot of flowering plants where birds used to get attracted. Over a period of time, she saw the population of birds visiting the her park came down. And she was trying to relate as to what could be the reason for such a decrease in the number of birds that was visiting her park. And that was felt all across as well. It was not restricted only to her backyard. Then when she was trying to relate to the cause, she could identify usage of chemical pesticides and their impact on the insects. And eventually, the birds which feed on insects also population came down, and that was very visible for everyone. So that became a kind of beginning of the awareness in the whole world. So Indian context, if you look at, we don't have much information, but we do have, but then it is not much. So I would briefly say the historical scenario of pesticide usage and ill effects in this environment, and then also give you a brief about as to what happened in North American European countries. And if you draw a parallel with the Indian scenario, where do we stand? Do we have very clear-cut case studies to say that XYZ species of birds suffered because of ABC chemicals? And if so, what are they? And then what in Indian context, institutions like Salim Ali Center for Ornithology and Natural History has done in the last 3-4 decades. And if we are very clear that these chemicals can create problem to birds, and what initiative that we can take towards mitigating the same. So this, what are the options that we have got in front of us so that we can tell farmers and try to use these options. So that is precisely the content of this talk. So first let me begin with the title slide. I guess many in the group, I do not know how many of you are having zoology background with little interest on wildlife. How many of you will be able to identify the species of bird which is on the screen? If I can just give 10 seconds to any of the participants who could identify, you can unmute and then try to make a guess. No. I guess none in the group is in a position to identify the species of bird what you see on the screen. I don't think you should feel bad because this bird almost now kind of extinct. And you might wonder as to why I show this bird. This is called Siberian crane. Used to be migrating into only India in Asia, only to Rajasthan in only to India in Asia, and then again only to Rajasthan in India, and subsequently, if you go further, it is only to Bharatpur in Rajasthan, and then again only to a small piece of wetland called Kiladu National Park in Bharatpur. This is a world famous wetland. It's a Ramsar convention site where we used to have thousands and thousands of migratory birds visiting this park. And this particular crane, Siberian crane, it's a magnificent, beautiful bird, about 5 feet tall. You should definitely see to appreciate the beauty of it. Unfortunately, over a period of time, the population kept coming down. I remember way back in 1986, when I was a research scholar, I visited this park. The first bird my seniors showed me was this, and it was really lovely to look at. That point in time, there were about 43, 44 birds. Over a period of time, the population kept coming down for various reasons. If you ask me as to whether it was because of environmental contaminants, I would say perhaps not, because during those days, our ability to understand or relate with environmental contamination was not much. So we had only one instance of one of these birds straying out of the wetland and getting, in the sense that it was found dead, but we could not identify as to what was the cause of the bird at that point in time. So precisely what I'm wanting to tell you is that this particular bird, as of now, is almost extinct. They have stopped visiting India. So like this, there are several species of birds which are facing threat, and they are under different category of getting endangered or extinct. So this particular bird, it is not that government of India or other countries where they belong to did not attach importance. They did attach importance. They breed on the river banks of Ob in Siberia, where the temperature used to be, still it is minus 80, minus 70. But then to tide over that kind of adverse climatic condition, these birds used to be migrating to India. But then, as of now, because of various reasons, we are not able to, have these birds visiting. Reasons are aplenty, because they migrate over various countries like Afghanistan, Pakistan, before they enter into India. There are several crane hunters who do hunt these birds. So that also could have been one of the reasons. But end of the day, what happened is that now we are almost, the government is ready to declare this bird as extinct in Indian context. So at this juncture, I also must tell you as to why I shared this picture as my title shared. I grew up in this ecosystem for almost 7-8 years. I should say that this is the aquatic ecosystem, the world famous wetland, which gave me that opportunity to research over there, and which eventually got me my PhD. So emotionally, I'm attached with this species of bird and also attached with this wetland ecosystem there. So that is one of the reasons why many of my talks, the title slide would be this. Well, now let us go into the actual part of the talk. So if you look at the pesticide consumption in this country, it has been on the rise. As I said, across the globe, pesticides are used for augmenting the burgeoning human population requirement. So that said, this has been the problem in the Indian context is more than what is there in the East, West, and European, North American countries, because the population increase here is more. So every year, what is available for cultivation, that is the land that is available for cultivation, is shrinking. So in that way, farmers are left with no option but to increase the productivity with available chemicals and other tools. So in Indian context, if you look at over the last 7-8 decades, the pesticide consumption has been very high. During that green revolution, scientists also recommended farmers to use chemicals because we were very clear as to how this use of chemicals could contain the pest and help farmers reap good harvest. So we had no way that we could have control on the farmers' choice of chemicals, use of chemicals. So that went uncontrolled for several decades. It was all in early 70s, we realized that yes, these chemicals have been creating problem. It was not because we did document or notice in Indian context, it was all because the Western world realized that these chemicals have created a problem to this environment, and birds could very clearly show that they are the best indicators. So on the whole, if you just look at, we use around 70% of the pesticides for agriculture operations and 30% for public health purposes. So if you look at the chemicals which had created problem, they all belong to a group called organochlorines. Chemicals like DDT, HCH, endosulfan, algreen, dialgreen, these are the chemicals which fall under the group called organochlorines. Over a period of time, when they created problem, they had to be banned. As of now, many of these chemicals, barring DDT, are not available in the market. They are not permitted to be used for any agriculture operations. DDT is the only chemical which, as many of you might know, was quite extensively used for containing malaria across the globe. Even till today, many South African countries where malaria is still prevalent, they continue to use large quantum of DDT for containing malaria through mosquito bites. So in Indian context, we do still use DDT to an extent of about 10,000 metric tons with the permission of the World Health Organization. But whether some amount of what is permitted to be used for containing, the mosquito bite, whether that is getting misused by agriculture for agriculture operations is a question which has been doing rounds, but we haven't got very clear cut answers. But then, based on the information what we have been gathering in terms of the residues in the environment, we do strongly suspect that there must be some amount of misuse as well, even till today. Unfortunately, chemicals that belong to organochlorines stay longer in the environment. They keep creating problems. You use today, 20 years, 30 years down the line, they are there still as residues in the environment, and then they keep moving up in the food chain and then create problems. So that is one reason that prompted the scientists and then which convinced the policymakers to bring a ban on many of these organochlorines, including DDT. Now, as I said earlier, in the international context, we had several species of birds whose population nosedived because of chemical pesticides, direct as well as indirect effect. In Indian context, until very recently, we never realized the fact that these chemicals can create problem to birds. Even when I began my career, there were several fellow researchers as well as bureaucrats, policymakers, always took it with what you call skepticism. They were skeptical about understanding it. It is mainly because they never wanted to believe, because that is going to have a larger impact on the agriculture economy. Now, if you're going to say that this particular pesticide is to be banned, definitely the companies which manufacture chemicals are not going to just accept it. So that has got a lot of ramification in economic circle. So that is one of the reasons why government of India also could not take immediate action. But then now you know that we are party to the Stockholm Convention, where we have signed, given a commitment that many of these organochlorine pesticides, which form part of the POPs, persistent organic pollutants. So that way we are committed to the globe that we shouldn't be using many of these chemicals, which are problem to environment. In that way, now we have brought in ban for many of these organochlorine group of chemicals, and then eventually we have started manufacturing, not only India, even in international context, many countries are now using largely organophosphates, carbonates, pyrethroids, and also biological means of containing this pest. In Indian context, we are little slow, but then again, now, as I said, we are constrained to listen to the world and also constrained to adhere to the commitments that we have given through signing many of these international treaties. In the Indian context, if you look at, it is not that we are suffering too much or perfectly safe, but then there are very many cases where we could very clearly say that these species of birds have suffered because of environmental contaminants, not necessarily because of organochlorines, but also because of the present day so-called safe chemicals, which belong to organophosphates, carbonates, and even pyrethroids. So if you just look at the list of birds which I have to share with you today, the first one is spot-billed pelican. The one that you see on the right-hand side top is spot-billed pelican, whose population has been gradually declining over the last 2-3 decades. There is a place called Kokravilleru. Perhaps I think I would come to that subsequently, but I would just go through this list quickly. Sarus crane and P4, over the last 2 decades, many instances of poisoning have been reported in many parts of the Western country. And Himalayan grey-headed fishing eagle, this is again a migratory raptor which has been unsuccessfully breeding in Carbet National Park in Uttarakhand. And then vulture population. We have lost almost 90% of the birds which used to be in Indian context. And they have been now left to few hundreds in certain pockets of this country, rather in its distribution range. Now I will quickly go into the details of these species of birds. First, let us begin with vultures. Vultures, they are called Karugu, more specifically Pranandani Karugu in Tamil, and in Hindi it is called Gir. So I think many of you will be able to understand. Plus, now I'm a little skeptical as to how many of you might have seen this bird. I don't know the age group, but then still I guess that many are below 30 research scholars. So that being the case, I guess you wouldn't have had that opportunity. But some faculty members, scientists who are participating might have seen, but then not in the last 1 or 2 decades. I remember way back in 1985, when I was given the task of surveying the species of birds in this entire country, we used to go really tired of counting these birds. Every 15 kilometers you travel, you have a small flock of 7,500 birds feeding on your carcass. But then now the scenario is totally different. So you may wonder as to what this bird is and what exactly led to the population decline in this bird. Let me quickly brief. So this bird is a scavenger. They feed exclusively on carcass. They are quite heavy and they fly up in the sky and they look for thermals and soar hours together to locate a carcass. The moment they locate a carcass, they descend and then feed on. So their population used to be quite enormous, super abundance in a way across the entire country, rather restricted to its distribution range. In the entire country, you don't get to see them in the same abundance because of various reasons, including food availability. Traditionally, in South, we haven't seen a large number of what you call huge size of, huge flock of these birds. Maybe 15, 20, occasionally up to 50 we might have seen, because the food availability is restricted in South India, because we know for sure that the cattle are not allowed to have natural death. So they are effectively used in this part of this country. But then in the rest of the distribution range, particularly Indo-Gangetic plains, their population used to be very, very large. Now the situation has reversed. Initial days when we started seeing these dead birds, we were trying to relate to pesticide poisoning, because back of our mind we know that pesticides have created problem in many North American, European countries. So we were trying to compare with that kind of scenario and trying to blame on pesticide. But then that point in time, we could not relate it perfectly. So because if pesticides have to be, then it has to happen through food chain buildup. We did not have adequate proof. Then poisoning is yet another reason. But that was also not possible to be proved. plus the analytical capacity, the laboratory establishment to look at specifically this kind of wildlife poisoning was not in place in Indian context. Though I started working right from 1987-88 onwards, the kind of facility that we had was very restricted, and we had to take samples to institutions where there were facilities, but then their area of specialization was totally different. The institutions which I used to go is Industrial Toxicology Research Center, Lucknow, but then their area of specialization is not wildlife toxicology. But nevertheless, they were helpful to me, and I also used to take my samples to Tamil Nadu Agriculture University, Coimbatore. I used to lug the samples all the way from Rajasthan to Coimbatore and then get them analyzed. But then it wasn't possible for us to prove that point in time that pesticide was the reason. Then veterinary scientists said that it could be a disease called gout. That was also not possible to be confirmed. Then looking at the geographical extent at which it was happening. What I mean is that this species of bird is not restricted only to India. We also have their distribution in Pakistan, Bangladesh, Nepal, Bhutan. So even there, their population was coming down, and scientists working over there also found a large number of birds falling dead. So we thought it could be a viral disease. Even that was not possible to be proved. Then another hypothesis came that it could be because of physical extermination. You may wonder as to what it is, if so, as to why one should do. Despite, you know that that these birds do feed on carcass. That means that they keep the environment clean by feeding on those putrefying carcass. So in a way, if you look at, I would say that they are Mother Nature's appointed scavengers. But then they also had a small nuisance value. If you recollect, I said that they soar up in the thermals. In the sky, they soar up because they, being a raptor, they have this kind of adaptation in the eye with which they will be able to locate the carcass on the ground only when they go up in the sky. So they used to be colliding with aircrafts, particularly fighter aircrafts when they fly at a low altitude with these birds. And then we used to be losing $1 million worth aircraft and also invaluable pilot life. So unfortunately, this nuisance value was bothering the pilots and also the common man, because we do not know as to who is going to be traveling by which flight and then which airport is going to have the hit of this. So it was also a major challenge for the flight safety authorities to handle this. So when I worked in this program, we also tried to find out as to how ecologically we can have a control on the population of this bird. But then incidentally, it so happened that there was some sort of intervention because of which many of these birds died. So as I said, we kept moving from one hypothesis to another. Finally, when we came to know that it was because of a drug called diclofenac, which is a non-steroidal anti-inflammatory drug, it was a huge surprise to the entire scientific community. So you may wonder as to what is this diclofenac, as to how this chemical created problem to this vulture. It's very simple. Diclofenac, as many of you might know, it's a painkiller. It is used even among human beings. You and me might have definitely had this when we had aches and pains. The first drug that a dentist would prescribe is diclofenac. Any rheumatologist would prescribe would be diclofenac. So any inflammation in the body, the first drug which any doctor would choose would be diclofenac. In the same way, veterinarians also quite extensively use this bird to treat inflammation in cattle. So if a cattle which had received a diclofenac dose just before its death, and subsequently when the carcass is eaten by vultures, and then residue remaining in the flesh will reach up to the bird, and then after exposure, between 3 and 7 days, these birds die. So this is how we lost millions of birds in this country. It was a huge challenge for scientists, managers to identify the problem and also find out as to how we can implement any conservation programs to bring the bird back into the normal quality. So now, if you ask me as to what we have done, I'll come. Before that, I would just show a few slides and explain as to how these birds were doing beautiful ecological service to this whole ecosystem. So now these birds, now you can see around 150 birds are sitting on the ground where you are not in a position to understand the carcass. They are there on a carcass, a buffalo carcass, actively feeding. So just see in another 20 minutes time, you would see the entire buffalo carcass left into heap of bones. This is the kind of service that these birds were doing. And if you just look at the kind of situation wherein you would need the services of bird, it's very obvious and clear. If you look at the Indo-Gangetic Plains, where the cattle population is quite high, and also the major source of revenue to the agrarian community is also dairy farming. So in a small village also, you will have 300, 400 households, you will have about 1,500 cattle. And then normal circumstances, without any disease outbreak, you will have 3-4 buffaloes dying every month. So there used to be families who used to be traditionally doing the job of collecting the carcass, bringing it to a common place, remove the skin, give it to a tannery, and then make money. And subsequently, when they want the bone, it is all deeply buried in the flesh. So then they look up the sky, vultures drop like bullets, matter of 15-20 minutes, they clear up the entire carcass and leave this bone behind. So these people used to go, cut into pieces, put it in a gunny sack, give it to a nearby mill, bone mill, make money. So this is how the nature was, in the sense that handling all the dead animals. And in the process, we ended up attracting vultures from the forest ecosystem into cities and carnivories. As you might agree with me, these vultures are not scavengers of our cities and carnivories. They are scavengers of the natural forest. So when carnivores like tigers, leopard, when they kill animals, they don't eat to the last bit. They leave a major chunk of their food. So that kind of situation, scavengers like vultures, even hyenas, they used to feed on those and then clean it up. But then now what is happening is totally reverse. You have carcass lying, putrefying, unheard. And you can see this area, it's not a kind of watertight compartment. Soon after rains, the entire area might get drained, and then the water might flow to the nearest water body, and where from the municipal corporation also might be supplying drinking water to the citizens. So it has got wider ramifications. So these birds can pick up these chemicals and then die. That is how it has been happening all these years. So you can see millions of maggots on the putrefying carcass. It is not only absence of vultures and putrefying carcass. So even, as I said, human health is related with this kind of situation. So now you may want to know as to what exactly the government has done or what the researchers have done. There has been a ban brought on use of diclofenac for veterinary purpose. So all the veterinarians have been advised not to use diclofenac, and then they also have been suggested to use alternate medicine called meloxicam, which is equally powerful, and in terms of its efficacy, it has been worked out, and then veterinarians have been advised to use that, and farmers also have been advised not to use this diclofenac on their own. Several farmers would start administering diclofenac on their own without even waiting for the veterinarians to come and treat the cattle. So now there has been a ban on this chemical. Plus now we also have several breeding centers across this country. There are several places wherein an institution called Bombay Natural History Society, with which I was associated till I joined the present organization, Salim Center for Ornithology and Natural History, has been coordinating setting up of this kind of artificial breeding centers in several locations. As of now, we have got in Haryana, Madhya Pradesh, Maharashtra, Assam, West Bengal. So these are the places we have got breeding centers where free-ranging birds are kept under captivity, encouraged to breed. Now a situation has come in where we are in a position to release the captively bred bird into the wilderness. So this is about vultures. Now let us look at the next species of bird. This is Sarus crane. I guess many of you might not have seen this bird. People in North might have seen, or still they have that opportunity to see this bird. They are birds associated with the agro ecosystem. You do find them in wetlands, which are part and parcel of many sanctuaries or national parks. But then largely these birds are found in huge congregations outside the national parks and sanctuaries, largely in the agro ecosystem. So they are pride of Indian wetlands. It's a wonderful sight to see a pair of dancing Saras crane, but when it dies, it's a pathetic scheme. This is the kind of situation that you would see in any wetland setup in the Terai belt. It's lovely to look at these birds in huge flocks and also in pair when they do the nuptial dance. It is really a celestial event, you should say. As you see, you might not be in a position to appreciate the size of this bird. It is said to be the tallest flying bird. Even man of average height has to stretch his body to match the height of the bird. This is a wonderful sight. You can see this bird sitting on its nest, where you can even locate a couple of eggs. So when the bird dies, as I said, definitely it is a very sad scene. Even you and me will not be able to match the height of the bird when you stretch. It's about 5.5 feet tall. That is why it is considered to be the tallest flying bird. This is one dead bird which we located in Gorakhpur in Uttar Pradesh. Well, now let us look at as to how these birds are suffering because of this pesticide. As I said, they largely are found in agro ecosystem where chemical pesticides are used. So in this, now otherwise also you would know as to how chemical pesticides are sprayed. Either it is with a handheld sprayer manually or with a power sprayer, as you see on the lower left-hand side, lower corner. But then the places where I documented large number of birds dead, the farmers had resorted to a novel technique where the farmer had got to the nearest dispensary, picked up a whole saline bottle, mixed his own concentration of chemical, tied it upside down. As a nurse would control saline input into the patient, here the farmer could also control the pace of this pesticide, which is to get mixed with the water, and then that mixed water used to be used for irrigating the crop. You may wonder as to why this practice was done. This was essentially to protect the crop from termite attack, largely at the sowing stage. So farmers used to even soak it overnight and then sow it subsequently. And also, till such time they are sprouted, they used to irrigate the crop, the field with this pesticide mixed water, so that the termites are handled effectively. So this is how many of these birds were getting poisoned in this area. So we know that circumstantial evidences that yes, this might be the probable cause. But then again, to prove scientifically, we need to definitely come out with concrete evidences. One of that is residue estimation. Residue estimation in the sense, subsequent to the bird's death, we need to conduct a postmortem and then take out all possible tissues, including the undigested food which is there in the crop, and then analyze them for various chemicals. So in the initial years, that was way back in late 90s, we had documented a chemical called dialrin, which was in high concentration aldrin in high concentration in the industry. So unfortunately, these chemicals, when they get into the environment, they also get metabolized and isomerized and then take two different forms. And many of the times it is more toxic than the parent compound. Here you could see the concentration of the aldrin, which was found in a high concentration, after getting converted into dialrin, is distributed to all the organs like brain, liver, kidney, and even in the adipose tissues in the muscle. So with this kind of information, we could very clearly prove that it was because of dialrin. So now dialrin or aldrin is banned because they belong to a group called organochlorine, which I said they are notorious. Because of that, these chemicals have been banned. And then now, if you ask me, are these birds safe after the chemicals ban? I would say no. About which subsequently I will say in detail. So when these birds pick up these chemicals, they show several symptoms. They keep themselves aloof, they starve, nodding and bobbing side to head. Head movements used to be noticed, started walk, violent flapping of wings with intermittent jumping. So this used to be the kind of symptoms which field biologists used to observe and then subsequently try to find out as to whether it is possible to collect these samples, send them across to the laboratory and get them tested. So this is one way we have lost a large number of these Saras crane population in the entire distribution range. We don't find this bird in the entire country. Their population is restricted to the central and western and portion of northeast. Otherwise, this population of this bird is largely found only in this restricted range in this country. Well, now there are lots of efforts being made. I hope these birds will not be pushed into a situation of what we saw in the case of Siberian crane. Well, the next species which I would say is spot-bill pelican. The population has plummeted from 2,000 to 330 within a span of 30 years. And then in spite of local villages opposed towards conserving the species, there appears to be less improvement. So the circumstantial evidences very clearly point out towards agricultural practices in this vicinity. Of late, in addition to these pesticides, we also have got nematode infestation, which has been taking a heavy toll. So there's a place called Kokhrabillary in Karnataka, where we have got a large number of these birds breeding in the village setting. So where the villages themselves are have been supporting this bird, but unfortunately their efforts have not been very helpful. But of late, in addition to the agricultural operations, we also have been having this disease as one of the reasons for this population decline. Then the next bird is peafowl, synonymous with dance, the national bird. I wouldn't say the population is coming down, but then they are getting poisoned across the entire distribution range, intentionally as well as unintentionally. In the recent past, their population has been on the rise, and then they are acting to an extent as a pest. So because of which farmers are little annoyed. As you know, these birds are found largely in agricultural ecosystem and also human settlements. So when they become pests, farmers think that poisoning them is the solution to get rid of them. So unfortunately, in the entire distribution range, we have been having large number of them getting poisoned. So the next species for which we have got adequate information is Himalayan grey-headed fishing eagle. This is a migratory bird and also a raptor. They breed in India. Other migratory birds, largely ducks, they come here only to tide over the adverse climatic conditions. Whereas this particular raptor, they come to India and breed and then go back with the family. Unfortunately, their population kept coming down, and then they also did not breed successfully. With specific reference to an ecosystem in the Corbett National Park, where we had this bird being monitored by one of my friends, who recorded these birds not to be breeding successfully. And when we had an opportunity to look at that unsuccessfully hatched egg, we had documented a very high concentration of DDT. 1973 ppm of DDT was recorded in the egg. So if I have to give you an indication, even if there is going to be 1 ppm of DDT in the egg, you can be rest assured that this bird is suffering because of DDT poison. So you may also wonder as to how DDT creates problem. Essentially, DDT interfaces with the calcium metabolism and does not allow adequate supply of calcium for the formation of the eggshell. Many of you might have heard about eggshell thinning effect. So the chemical involved in that kind of thinning is this DDE. DDT has got several isomers and metabolites. So one among that is this DDE, which is responsible for interfering with the calcium metabolism and eggshell thinning. So when the eggshell thickness comes down, parent bird, when it sits on the egg to incubate, the egg doesn't have the necessary weight to withstand the weight of the bird. So then it cracks, and sometimes it does not even hatch properly. Or even if the egg by hook or crook hatches, the chick that comes out will have several deformities. This is one reason why this bird did not breed successfully, because of which the population also came down over a period of time. In addition to many of these flagship species which I explained, many of the so-called common birds population is also on the decrease. One is this drongo, house sparrow, Indian roller, Baya weaver bird. These are some of the birds which we used to see in large numbers in agro ecosystem, are now not in that kind of concentration. The reason are aplenty, but we cannot relate only to pesticides, but then we have every reason to relate pesticides because these birds are found in the agro ecosystem. When I was here, nothing which you can see these days so easily. There are places where you can see, but then the population is on the decrease because they feed, their major habitat is agro ecosystem. So their food base is reduced. Their landscape, land use pattern has changed. So that is one reason why many of these birds' population is on the decrease. Well, now with this kind of background information, in the national level, we have been following up the impact of these chemicals on the birds using birds as an indicator. So we have done this kind of study, or we have been involved in doing this study for the last several decades. So over a period of time, we have received several dead birds in our laboratory, and the number of birds might have been maybe 2,000, 2,500 birds, and number of species also has crossed 125. So if you just look at the birds and relate it with their feeding habits, all the birds, as you know, don't feed on everything that is available. So they have their selective feeding choices. So some birds feed exclusively on insects, we call them as insectivores. Some big birds feed on fishes, though we call them as fish-eating birds. Which eat everything is called omnivore, which eat only flesh is called carnivore, and birds which feed on plant materials is herbivore. So that way, if you look at the insectivorous bird seems to be the worst sufferer because of these pesticides. So now let us look at in the recent past what has happened. So even birds which are there in the aviaries, zoological parks, they are not safe. You may wonder as to why birds which are then kept in the zoos or biological parks are suffering, because the food comes from outside, where the vendors or the contractors, they get the bird being fished from the agricultural, rather the aqua farms, or even in the natural settings where agricultural drainage join or intentional poisoning happen. So subsequently, these fish, when they are fed to these birds which are there in the aviary, definitely they pick up the contamination and show up. Like that, now we even had such a problem in Vandalur Zoo a couple of years before. Then house crow, you may call it as a most common bird, but it is, yes, it was common, and maybe in certain areas still it is common, but then large scale mortality keeps happening, and intentional poisoning also happens. In some places it is being, they are being hunted for flesh, which is being sold in the market as chicken. So that is one reason why these birds are getting poisoned in large scale. And as I said earlier, people in the entire distribution range, we are having them getting poisoned in large numbers. Even last year, we had several of them dying in Nagarkoil, Tirunelveli belt. And the Madurai bench of the Madras High Court took suemoto cognizance of this instance, and then they directed the forest department to collect the samples and submit to our laboratory, and then give the report. We very clearly said it is because of poison. And the next species which we have seen in large numbers dying because of chemical is domicile crane. This is yet another migratory birds which we used to see them in thousands. You need to believe that migratory birds, when they visit the western part of this country, miles together, you will see them sitting like this. So definitely you will be amazed. It's a spectacular scene. You need to see to appreciate the kind of concentration that birds have, particularly in the western part of this country. Last year, I was there for a couple of days in Porbandar. I could not believe the number of birds that I saw. They are also getting poisoned. Farmers don't intentionally poison, but then year after year it has been happening. And then if you look at the reason, it appears to be because of pesticide. In a couple of situations in Gujarat, we had this opportunity to collect these birds, and we documented high levels of organophosphates and then even pyrethroids in these birds. And in this particular case, we found 4A to be responsible. They are granular form of pesticides. Farmers used to mix along with the seeds when they sow. That is again to protect it from termite attack. Incidentally, these birds voraciously feed on the grains also. When they pick up these grains, they get poisoned. So some of the symptoms were very clear in terms of indicating that they are because of pesticide poisoning. The symptoms are like excessive salivation, foam around the mouth, breathing difficulties, weakness, inability to fly, teary eyes, runny nose, and also they get the paralytic attack. So with this, we were able to understand that it is because of this porate. And additionally, we also documented rodenticides, which are used for containing or killing rats in the agricultural field. And they don't allow the blood to clot. The moment any rodenticide is picked up by this bird, and when it reaches the system, the bird is susceptible to bleed and die. If there is going to be small rupture somewhere, you can see on the blood on the left-hand side, the bird that you see on the left-hand side corner. Almost all the 3 slides, you can see the blood oozing out. So like this, we lost several of them. So this is yet another reason as to how these birds can be used as an indicator to understand as to how these chemicals are dangerous to the entire environment. In addition to that, we also have had several instances where migratory ducks like poachers, moorhens do die because of this pesticide. Even swift, we had a rare instance where DDT was proved to be responsible, where around 150 of them died near Madurai. So now, if you ask me, is that only birds are suffering? Many of you would agree with me that it is not. You and me are also the sufferers. Only thing is that Mother Nature has given us some sort of capacity to assimilate, withstand, put up with this kind of residues which we ingest accidentally. But then eventually, if you look at, they have got several ill effects which are quite extensively documented in many countries. Certainly, human beings are suffering, and certain areas where it is documented, and largely it goes unrecorded. The one that you see on the screen is a clear indication as to how people picked up this problem. This particular slide belongs to a place called Padri village in Kasaragod district in Kerala, where endosulfan had created a lot of problem among people, like cerebral palsy, epilepsy, miscarriages, and even cancer among people. But then immediately government could not act. What exactly had happened was the Kerala Plantation Corporation had extensively used a chemical called endosulfan, aerial. They used to be hiring choppers and then spraying this unmindful of the situation on the ground. And there are several schools, villages, and human settlements, water bodies, unmindful of all that they had used extensively, which led to greater population. Affect. And of all the places in Kerala, and in the country, Kerala turned out to be a location where we had extensive extensive problem which was felt among this poor. And the earlier government had constituted a committee wherein I was also one of the members. We collected samples of blood of people working in the plantation sector, even mother's milk, and documented high levels of this residue and gave a report to the government saying that endosulfan appears to be a culprit chemical. Even the industry which was manufacturing endosulfan intimidated the scientific community. They wanted SACON to particularly me to withdraw the report. We said, no, this is a scientific finding which cannot be withdrawn. And if they want to challenge, they can challenge in their own way, either in the court or wherever that they think is apt. But what I am trying to tell you is that chemicals have created problem to human beings, and we have got lots and lots of situations. Even in Punjab, if I don't know how many of you might have seen the program which was hosted by Amir Khan titled Satyameva Jayate. So several birds had, several people picked up this problem of cancer in several villages in Punjab. They used to be coming to a hospital in Rajasthan by train. Because of large number of people traveling by this train, the express train itself was called as, by locals or identified as Cancer Express. So people go to the counter and then ask for a ticket by Cancer Express. That is the level of prevalence of this problem of cancer in that village. So elsewhere also it has been happening. But only thing is that doctors do not try to find out as to what can cause this cancer. They go symptomatically. It is like now, then you pick up any cough, cold. No doctors would ask as to how did you pick up this. It is instantaneously prescription is given. So same is the case for cancer also. Every doctor would look at worst comes, they might ask as to whether someone in their family had this. Otherwise, it is chemotherapy, radiation, surgical intervention, all that. That's a typical protocol. And then they may declare that 10 years, 15 years of lifespan. But then they don't look at the reason. We cannot find fault with them for the simple reason that they are not taught to find out the reason. They are taught to treat the patient. That is how it is. Now, if you ask me, having understood that this pesticide problem has been felt extensively across this country, and also people are not exemptions. It is not only birds which are suffering, even human beings have been now documented to very clearly face the problem of pesticides. But then now we cannot go back to farmers and then say that don't use pesticides. And they have to be told as to how they should contain this pest on the crop. Otherwise, we need to definitely cater to the people's food requirement. So, as I said in the introductory remark, that the available land is shrinking. So, they have to be given options to enhance the productivity within that land that is available. So that way, farmers are also constrained to use chemicals or look for tools which will help them to augment the food production. So, nevertheless, we also have got certain tools which we can suggest to farmers. These are all not the inventions of me or SACON. These are all inventions of agricultural scientists. When they were forced to find out alternate ways to handle the pest, they had to look at other available options. This integrated pest management program is the one which looks at kind of in the ecological way. So they try to find out as to how the use of chemical can be minimized towards containing this pest on the crop. Then the second thing is a good agricultural practice. What is this? This is essentially advises to use chemical when it is unwarranted. Sorry, when it is warranted. So farmers have to be taught as to when they should intervene. Every plant has got its own capacity to withstand, like what we call it as economic threshold level. So only at that point in time, farmers also have to intervene. Otherwise, we don't have to bother at all. But then we are tuned to immediately act. So in a way that we, without understanding the threshold of the plant, we immediately apply pesticide and end up creating more load on the environment and then eventually leading to other cascading impacts. So the next option is to organic farming. This is the kind of eventual tool that is available to all of us. Across the globe, if you look at, there has been larger acceptance, and governments also have been promoting this organic farming, which recommends absolutely in the sense that we cannot say that this is not supposed to be, in the sense that cannot be accepted by farmers. In the initial years, there may be resistance because there may be loss of productivity. But eventually, over a period of time, when the soil regains its value, health, the plants will definitely do good. So, over a period of time, farmers will also understand with less input, they can reap better harvest, leading to greater economic return. So, this requires a lot of creation of awareness among the farming community. So, to which government is putting an effort. There are a lot of encouragements. Even we were part of Kerala government when they wanted to promote this in Kerala, and states like Sikkim, they have an organic farming policy. In Sikkim, there is no pesticide at all. So like that, now many states are trying to encourage farmers to use less chemicals or resort to organic farming, whereby we will be able to handle the impact of contaminants on the environment using bird as an indicator. And I think I should stop here, and thank you so much. This is the institution called Salim Ali for Mythology and Natural History, where I work. This comes under Government of India Ministry of Environment and Forest. We do work on nature conservation, environmental protection across different landscapes in this country. We have various research divisions like avian ecology, conservation biology, landscape ecology, wetland ecology, environmental impact assessment, ecotoxicology. That is the division in which I work. And we also have a division called avian genetics and physiology, nature education and extension. There are these are the 10 divisions in which we have scientists working in different parts of this country. Our headquarters is at Coimbatore. It's called Anaketi, which is about 30 kilometers northwest of Coimbatore city. We have 50,000 square feet built-in area wherein we have the administrative, laboratory blocks, library. So this is about SACON and the work which we have been doing. So if the participants have got any questions, they can feel free to ask me. Thank you so much. I take this opportunity to thank the Avinashilingam University for having chosen me to give this talk today. And also I thank all the participants for having enrolled for this program and having listened to me. And if you have got any questions, definitely you can ask me. If not today, subsequently also, you have got my email address, I guess. You can definitely communicate with us. And you can also help us in terms of executing this work, in the sense that you can chip in your efforts. When you have your dead bird in your area, you can just give us a call. We will try to find out as to how we can get the bird sent to our laboratory and identify the reason for the mortality. If it is pesticide, we can find out as to what exactly is the chemical, and then what is its usage pattern, what is the policy that is in place, whether it was illegally used or illegally used. So all those things will help us in terms of handling the issue in this country. Thank you yet again. And over to the organizers to facilitate if there are any questions among the participants.
Thank you, sir.
Yeah, my pleasure.
Very informative session, sir. Now the forum is open for discussion. Participants, you can just post the questions in the chat box, or you can just unmute and ask the questions.
Are there any questions? Is it that the talk was self-explanatory, so you have no doubt, or it was so boring that you did not have any question to ask on that? I don't know which one I should consider. Should I sign off?
Any questions, please, audience?
If there is no question, shall we move on to lunch break now?
Thank you, sir.
Okay, thank you so much. So I'm
Thank
signing
you,
off.
sir.
Okay, I'm signing off.
Yes, sir. Ma'am, shall we move on to lunch break, ma'am?
Yes. The audience joined by 120.
Now we'll have a lunch break, and please join the next meet on 1:20 p.m.
Good afternoon, sir. Why it's blocking? Ma'am, sir has come.
Hello, ma'am. Can you hear me, ma'am?
Yes, sir.
Okay, wait a minute.
Yes. Good afternoon, one and all present here. Now we'll begin the technical session by introducing Dr. M. Tangarasu. He has core competency like hands-on experience of machine learning algorithms, knowledge and experience of natural language processing, and proficiency in computer and IT systems, and key skill in scripting, programming like Python and R, tools and packaging, database like MS Access and Oracle. He has work experience of about 8 years and completed projects like computing approach to natural language processing for categorizing massive Tamil scalable data, healthcare informations, extractions from image dataset using named entity recognized with NLP, and so on. He has participated in several conferences and workshops and published 5 papers in reputed journals. We are very fortunate for having you here, sir. Over to you, sir.
Hello, can you hear me?
Sir, we can hear you, sir.
Ma'am, ma'am, make me my screen as presenter, ma'am. I will share the screen.
Okay, sir. Sir, now you can present the screen, sir.
Wait a minute, ma'am. Are you able to see my screen, ma'am?
No, sir.
Hello?
No, sir.
Wait a minute.
Now it's visible, sir.
Okay, ma'am. Okay, I'm audible right now?
You're audible and it's visible, sir.
Okay, thank you. First of all, good afternoon, everyone. I thank Avinashilingam University for giving me this opportunity to present my knowledge to here in this session. Basically, in this session, we discuss about statistics for data science and business analysis. First of all, statistics is used to mainly to gain an understanding of the data and focus on various applications like regression analysis, predictive analysis, and forecasting analysis, interactive data visualization. Then it is a process of collecting data, evaluating data, and summarizing it into mathematical form. Initially, statistics were related to the science of the state, where it was used in the collection and analysis of facts and data about a country, such as economy, population, and etc. Then it's into mathematical statistics, applies mathematical techniques like linear algebra, differential equation, and mathematical analysis, and theory of probability. This is the general introduction about statistics. Why statistics in data science? Statistics is at the core sophisticated machine learning algorithms capture and translating data patterns into actionable evidence. Data scientists use statistics to gather, review, and analysis and draw conclusion from the data, as well as apply quantified mathematical models to appropriate variables. This is the importance in statistics in data science. We can classify the data statistics into two categories. One is descriptive statistics and inferential statistics. After that, we discuss about inferential statistics. I'm not going into deep into the descriptive statistics because we have only 1 hour time. So, descriptive statistics. So, method of statistic is used to describe the data collected and summarize the data, and it is properties is used the measure of central tendency and the measure of dispersions. And inferential statistics, this method of statistics is used to draw conclusions from the data. It requires statistical test performed on samples, and it draw the conclusions by identifying the difference between the two groups. For example, test calculate the p-value that compared with the probability of chance alpha equal to 0.05. If p-value is less than alpha, then it's concluded that the p-value is statistically significant. And the descriptive statistics classified into two categories: measure of central tendency and measure of variability. Tendency is mean, mode, median, and variability, it's range, variance, dispersion. The important thing is in Python language, for statistics we used pandas. It is a very efficient package. It providing fast, flexible, and expressive data structures designed to make working with relational or labeled data both easily intuitive. It aims to be fundamental high-level building blocks for doing practical and also real-world data analysis in Python. The importance of the pandas is we easily handle the missing data and size mutability. Size mutability means we can edit or delete or update values using pandas. But if you use tuples, we can't mute the values. Then another one feature is automatic and explicit data alignment. We can see about these features after that. And another one is we can easily split, apply, and combine in data frames values. And the most important features are slicing. Slicing means we can randomly choose the particular column values. And fancy indexing and subsetting. Merging and joining is more efficient in using pandas. For statistical analysis, we go with pandas. We can easily handle the data. It is also support the SQL. SQL also it support. And also flexible to reshape and pivoting of the data set, and also hierarchical labeling of access. We can informative access we can used in pandas. Pandas is an efficient package. It can handle the CSV files, XS files, JSON files, HTML files, SQL files, and pickle files. We can compress or decompress the data. Also, it's supported. In pandas, it's all based on the rows and columns like tables. This is the general introduction about pandas. In additionally, it has the border of goal of becoming the most powerful and flexible open source for data analysis. It handles the different kind of data like tabular, CSV files, Excel files, JSON files, and all. Why we use the pandas? Pandas is the fast. Many of the low-level algorithmic bits have been extensively used in Python code. These are all about a general introduction about pandas. Then how to import the pandas in Python? Just you can give you install the pandas from Anaconda Conda environment. Then we can import pandas as PD. That's it. And if you handle the array, we can use NumPy. Just you can import NumPy as NP. For visualization, data visualization, we use two kind of packages. One is Matplotlib and Seaborn. For this analysis, this tutorial, I take Titanic dataset for analysis. I used two inferential models. One is logistic regression, and another one is grid search CV. Logistic regression based on the linear model, regression model, and grid search CV is the one of the best classification algorithm in machine learning. So I compare the two algorithm, and after analyzing this algorithm with the confusion matrix. This is the agenda of this tutorial. First, we can take the Titanic dataset. In this Titanic dataset, these are all the columns. In general terms, in table it is columns. In the data sense, it is a features. Passenger ID and survived. P class, name, sex, age, and these are all the features. How can I load the local Titanic dataset into in pandas? See, it is syntax for the read the CSV to pandas. It is just a variable. Titanic is just a variable. We can change the variable. And PD, we import the pandas, right? Pandas as BD. Look, BD dot read CSV, and file path. Once you run this query, run this column, now we can run print the Titanic. Look, just we can read the CSV file from the local computer. Look, pandas gives this much of information, sir. Look, here 891 rows and 12 columns. These are all the column names. Index start with 0, so 890. So total is 891 rows and 12 columns. Passenger ID 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 columns. Okay. Now we can we have a data frame as Titanic. Now I need only this head option, head operations. These are general statistic analysis from the data science. Suppose if I fix a Titanic dot head 10, I execute this. Look, 0 to 9. That means 10. 10 records, 10 rows. It's displayed. Suppose I need 15. Okay, it's display the 15. Clear? Suppose I need tails. Wait a minute. Okay, I'm not confusing. Okay, it's display the 5. And suppose we need between the range, we need to display the records. We can use index locations. Titanic dot index locations 25 to 30. It display the 25 to 30. Suppose we need to change 30 to it display the 30 to 35. Then generally we calculate the mean. Use in other languages we can use some other technique. Some other we can calculate the manually, or write the code manually, or anything. We just here we just give wait. Suppose I need the column age the mean of the age column. Just you can give the I think I lost the connection. Wait a minute. Sorry. Sorry, I lost the connection. Oh. Just you can give the Titanic age dot mean. It gives the mean of the age column. Clear? Then if you need to calculate the median of age and fare, just you can give the Titanic age and fare dot median. it gives the median of age and fare, and also give the which kind of data type. 28.00 and 14.45. It is a float type, so it gives the float. Then another one important feature is describe. Describe means suppose you give the need the age and fare, we can describe using describe function. It gives number of counts and a mean, standard deviation, and minimum and 25 percentile and 50 percentile, 75 percentile, and a maximum. This is the use of the description. And this aggregation function, we can aggregate the columns. Suppose we give the age and fare, we need only minimum, max, median, skew. So you can give this age and fare, minimum, max, median, skew. It gives the minimum, max, median, skew, mean. These are all the basic statistics. So I can present here. And another powerful is group by. In this group by, suppose I need mean of the sex and age. We can give the Titanic sex and age dot group by sex dot mean. It gives, it is the age, this column age, this column sex, female. The average, the mean of female is 27 point something, and male is 29. And another one is just you group by the sex. You can give the Titanic dot group by sex. That mean it gives passenger ID and survived, P class, age, siblings, this information, and female, male. These are all the mean. Like, actually, it is are all the simple questions. I work out these questions, and these are all the results. If it is a hands-on session, it is very helpful. Then we generally know how the how we can analyze the data and visualize the data. These are all the basic techniques. Suppose we need to group by fare based on the sex and class. In Titanic, we have three classes. Class 1, class 2, that means ticket classes. That in flight we have economic class and business class, right? Like that, it is three classes: lower, median, and high. Like class 1 and class 2 and class 3, it is the mean of the females in class 1 and class 2 and class 3. It is a male mean of the class 1 and class 2 and class 3. Look, these two columns, here it is shorted. The second one is it gives the result shorted one, 1 2 3, like it generally gives. Suppose you give the Titanic class dot value counts, it gives which one is highest first, like that it gives based on the highest list. It gives. Suppose you can use the Titanic dot group by T class dot count. It gives shorted one. That is a major difference. See here, I assigned one variable to Titanic. We have a data from Titanic and we transfer the data into train. Then another one important feature is heat map. Heat map contains the contains value of representing various shades of the same color for each value of the value to plotted. Here, to get the column which has null values, I choose the column value which column have null values. In these 12 columns, age and cabin has null cabin, and this last column has null values. This yellow one, right? These three columns has null values. This is the heat map feature to visualize the data. Now we are entering into the visualization. How to we present the data. Before we seen about basic statistic analysis. Now we enter into the how to show our data. Look, this SNS is Seaborn. Already I told we have import two packages. One is Matplot and one is Seaborn. This feature support the Seaborn package support these kind of visualization technique. So SNS.count plot survived data equal to trade. Zero means not survived. One means survived. If you see the description of the data, the survived, this feature have value 0 and 1. 0 for not survived and 1 for survived. When you see this this graph, this bar charts, we know 0 means not survived. More than 500 passengers are not survived, and near 350 360 patients are not patients, passengers are survived. And how many males and how many females? Survival based on the sex. sns.count plot, x equal to survived and hue equal to 6, data equal to train plot. We can use red and blue. Blue graph means male and red means female. See, more than 500 are not survived. Near 450, 460 male are not survived, and 80 near 80 females are not survived. And survived males near 110 males are survived, and females near 250 females are survived. This is the visualization. Visualize the data. How to we represent the data? Represent and present the data. Like this is survival based on the passenger class. Blue bar chart bar represent class 1, passenger class 1, and orange represent the passenger class 2, and green represent the passenger class 3. These are all the simple visualizing technique, and based on the class we can easily present the data here. And another the first we seen about general statistics, mean, median, mode kind of things, and other we can show the seen the bar charts and relations between the two columns. And now it is a histogram. It is another kind of visualization technique. Look, SNS displot, train, age, drop null. Drop null means if data have null value, it's drop the null values and it shows. And also this one, out based on a sibling or spouse. Here, look. Another one is this pins. We can adjust the graph. Look, wait a minute. Now here I give the 40. Suppose I give 10. It's adjust the histograms. You can see the changes. And recent days for visualization in data scientists, we use the Plotly. It is more efficient visualization package. Just you can import the install the cufflinks, and we can use the Plotly. what is the difference between these two graphs? Suppose we can navigate this. See my cursor movement, it not display anything. Here it gives the information about this for. This is the major difference, major advantage using a plotly. Another one visualization techniques is box plot. This center black code, you see the center it is the highest value between the 30 to 40. That means 35. There are more number of cases. This is the box plot. And generally I written on functions, on function that impute age, and this impute age, here I call the function impute age. We taken two columns, age and p classes, so we pass the column. Suppose age column, pass the column 0 and 1. If it is null, value, if p equal to 1, it return 37. We generally assign the impute the values. How to impute the values? This function for inputting the values. Look, the heat map. Once we impute the ages, look the heat map. Compare this on the this heat map. We can only two columns have null values. Look, now age have we inputted the values into null columns. Look, compare this heat map and the previous one. Look here, these two columns have has null value. Three columns has null values, and age also has null values. When we input the values, we remove the instead of null values, we use the here this function. Look, this is the power of imputation. Now from train, wait a minute, I will show. This is the train data. We have data frame 12 columns. When I drop the cabin, how to drop a particular column in the train? We compare these two. Compare these two data frame. Here we have a cabin. We removed the cabin. Look here. Here sex equal to male and female, right? When we use the get dummies function, is used to convert the categorical variables into dummy indicator variables. That means, suppose, look, 6 equal to bd dot get dummies. We have strings, right? Male and female. Here, look, we have a male 1 and female as 0. Generally, we have a string in your 6 column. Now we can convert it into strings to categorical variable. That means 0 and 1. We in the column we have only two values, whether it is a male or female. So we use the get dummies functions. We can easily convert into categorical variable. This is a use of get dummy functions. Another one we can also like that from the get dummies functions impact parked we can convert it into categorical variable. See? Then we need to the compare the two models. One I already told when I before start the session. I told we have we'll see the two kind of inference model. One is this one regression, logistic regression, and another one machine learning model is grid search CV. Now we need to train the data. For training data, here we drop the sex, impact, and name on ticket. And now our training data only have this one: passenger ID, survived, P class, age, siblings, watch, and fare, male. Instead of using 6, we have used S, and impact means Q. Compare these two data sets. Here, look, some columns have strings and all string values. When you need to train your model, we need only numerics. See, This data set now converted into full of numeric. Now we enter into this one inferential models. See, first we seen about what is statistics and why statistics, and another thing the basic statistic analysis and how to visualize the data, and how we can change the our data into categorical variables using get dummy functions. Before we discussed these three stages, after that we enter into inferential algorithms. See, now we can enter into we need to take our training and testing set. We only drops survived and stored into x variable, and survived we store into y variable. Then I split it into x10, x test, y10, and y-test equal to using training split. We have a data set, data frame x and y. So I'm taking 0 point as testing test. That means 0 point means 0.3 means 30% of the data we can use for test. Wait a minute. running out of time. Generally, regression analysis is set of statistical method used to estimate estimation of relationship between the dependent variable and one or more dependent variables. It is a simple regression. A simple linear regression, y equal to mx plus c. It is a very basic method. We put on a slope, and we can adjust the slope, and we can classify, or we get the targeted output. And logistic regression, it's a weight based. Wait a minute. This logistic is a statistics model that in basic form uses the logistic function to model a binary dependent variables. We can get in linear regression. We can get get multiples of inferences. Here we get only binary in logistic regression. Whether in this case, suppose we pass the features or weights, the model can able to give binary out, whether it is happy or sad. Maybe it survived or not survived, like that. Look, I load the linear regression model. It is a pre-trained model from these sklearn packages. For data science, the sklearn package is very important package. It gives multiple models, machine learning models. I load the model, logistic regression, and fit the X-train and Y-train. We splitted the training, right? X-train and Y-train. Actually, generally, if you work on this hands-on, you clearly understand these concepts. If you start the research, we need to compare two models, right? From that comparison, we can get one better result. So this logistic regression, I am using the Titanic data set. It gives what happened? Connecting very slow. See, we get the prediction. We take this data set, not survived 154 and survived 114. It predict 85% accurately for logistic regression. This is the confusion matrix. If you're familiar with the confusion matrix, you can easily understand this. I generally give, look, true positive, false positive, true negative, false negative, these things. The thing is, we can get the F1 score for class 0, it is 82%, and class 2 is 72%, and overall accuracy is 78% for logistic regression. And machine learning model, I use a grid search CV. Here I import the grid search CV from sklearn. In grid search CV using I'm random forest classifier. Same data set, I'm trained the model and I fit the model here. Look, compare the it gives overall accuracy is 80%. F1 score also improved 84% and 74%. This is the confusion matrix. So we can conclude from these two analysis. From this analysis, we conclude one thing: compare than the regression model, this grid search CV model give better results. Yeah, first thing. First, we can see the introduction about the statistics and which package for app in for using the statistics in data science. It is a pandas data frame. Why we are using the pandas data frame? We can handle the multiple files like CSV files, Excel files, JSON files, and HTML files, SQL files, and pickle files. We can easily compress and decompress the files. So we can go with pandas framework. Then for our analysis, we take a Titanic dataset. From this Titanic datasets, we can apply the basic statistical functions, and we cleared this one, the how to present the data in pandas, and we enter into the visualization. We see the heat map, histograms, and bar plots, and this one, histograms, box plots, and we enter into inferential system for the Titanic dataset. We seen the logistic regression model, how to load the logistic regression, and we fit the model, and we generate the confusion matrix. From this confusion matrix, the overall accuracy of logistics model is 78%, and a grid search CV, that means we used here random forest. In grid search CV, we have used the random forest classifier. It gives 80%. From this analysis, the better model is grid search CV. That's it. Thanks, ma'am. Thanks for giving this opportunity. Please, any queries, reach out me in tangrasamadhan at gmail.com. Time it's out of running. Sorry. Hello, can you hear me?
Yes, sir.
Yeah, that's it, ma'am.
Thank you for your informative session, sir. The session is open for discussion. If you have any doubts, please post in the chat box.
Yeah. Ma'am, doubts, if you have doubts, please share, send to this this mail ID, ma'am. I present the mail ID, right? If you have any doubts, anyone from computer science or any other, because these models not only for computer science, we have take different kind of data sets, whether gene data, any other data, botany, we can convert it into encoded the data, and we can use these models. This is special in data science. Whether we have used it in healthcare or any other domain, we can use this model. But we need to fine tune the model. That's the thing. If you know how to fine tune the data and all the things, we can easily understand the machine learning and this one deep learning and data science. That's it, ma'am.
Okay, sir. Participants, if you have any doubts, please mail it to sir's mail ID. It is there in the presentation. Now we'll be moving on to the next session. Thank you, sir.
Thank you, ma'am. Thank you for giving this opportunity.
Good afternoon, sir.
Now we will begin the technical session by introducing the resource person, Dr. Kannan C.S. Warrior, Senior Principal Scientist at the Institute of Forest Genetics and Tree Breeding, Coimbatore, of the Indian Council of Forestry Research and Education with the Ministry of Environment, Forest and Climate Change, Government of India. He worked as a consultant member to Andhra Pradesh Forest Development Corporation and Kerala Forest Development Corporation for raising high-yielding colonial plantations. He received the Rolla Esrau National Award by the Indian Association of Angiosperm Taxonomy for the best research work on biodiversity conservation in India in a non-forested region in Kerala. He has 27 years of experience in forestry research. He has published 268 research papers. He delivered 239 awareness lectures on various topics, functioning as a coordinator of Envis Center on Forest Genetic Resources and Tree Improvement of Government of India, Nodal Officer of the Green Scale Development Programs of Ministry of Environment, Forest and Climate Change, Government of India. We are fortunate to having you, sir. Over to you.
Good afternoon, sir. How are you, sir? Sir, please unmute your mic, sir. Yes, sir. Yes, sir.
Namaste.
Namaste. Sir, how are you, sir?
Thank you. Thank you.
Sir, Dean Ma'am is there.
No, thank you. Oh, hello, ma'am. Namaste.
Sir, can we start, sir?
Yes, ma'am. Yes, ma'am.
Yes, sir. Yes, sir. Sir, we would like to have your song, sir.
Sure.
As usual. Yes, sir.
Sure.
Even the participants will be delighted to hear that.
Thank you, ma'am. Namaste and good afternoon to all. This lecture is on nature. We shall begin this lecture with an invocation on nature, a Prakriti Vandanam, based on Yajurveda, composed by myself in diverse ragas of Carnatic classical music, Nata, Ananda Bhairavi, and Hamsathani. This was released for the Ministry of Environment, Forest and Climate Change. A Prakriti Vandanam. Sakyam Devadatma Himalaya Dhyeyo Raivatako Vindhyo Girischaravalistadha Girischaravalistadha Om Dho Shantirantarikshaha Shantihi Prithvi Shantirapaha Shantiroshadhayaha Shantihi Vanaspadaya Shanti Vrikshvam Devaha Shanti Brahma Shanti Sarva Shantihi Shanti Reva Shantihi Sama Shanti Redhi Om Shanti Om Shanti Om Shanti
Thank you, sir.
Most welcome, ma'am. I shall
Yes,
share
sir.
the
Yes, sir.
presentation.
Thank you so much, sir.
I shall
Sir,
share
you
my...
can, yes, sir. You can start.
Yes.
The
Yes. Is it visible, ma'am, now?
sir, yes, sir.
Sacred groves are patches of native vegetation protected on the basis of religious faith. Apart from India, they occur in different parts of Asia, Africa, Europe, America, and Australia. In India, sacred groves are mainly distributed in the states of Andhra Pradesh, Karnataka, Madhya Pradesh, Odisha, Maharashtra, Rajasthan, Uttar Pradesh, Tamil Nadu, Kerala, and northeastern states. Most of the states, sacred groves are present. Sacred groves are one way of expressing our gratitude to nature that supported our life. And they are protected to avoid the perceived wrath or anger of the resident deity. Sacred groves vary in size from few trees to dense forests covering vast tracts of land, and it's been estimated that around 1 lakh to 1,50,000 sacred groves exist in India. At present, sacred groves are not merely patches of forest but are islands in desolated landscape. Many valuable medicinal plants and wild relatives of cultivated species are present in the sacred groves. They got tremendous role to play in any species improvement or tree improvement programs. The role of the sacred groves as micro watersheds is also of considerable importance. Many streams originate from the sacred groves. Though our ancestors understood the importance of the sacred groves, at present it's been forgotten due to the lack of accurate and adequate information. According to the National Environment Policy in 2006, it says that the ancient sacred groves should be treated as possessing incomparable values. It means, like we protect the valuable monuments like Taj Mahal and charismatic species like lion and tiger and valley of flowers, we have to protect the sacred groves. We have to consider sacred groves which have got incomparable values. Sacred groves are known by different names in different parts of the country. In Tamil Nadu, they are called as Kovil Kada, Karnataka Devara Kada, Maharashtra Devarai, Than is a name in Assam, Serna in Madhya Pradesh. In Rajasthan, sacred groves are called as Orans. In West Bengal, they are called as Garimda or Gramda. In Himachal Pradesh, they are called as Devan, and Locking Packs in Meghalaya. More than 90% of the Meghalaya's forests are owned not by the government but by communities and individuals. Around 68 groves have been reported in Meghalaya, and many streams originate from the sacred groves. For example, for this Lam Shenglong Nokrin sacred grove, for example, is a source of 8 streams that supply water to Shillong. Floristic diversity studies conducted in the sacred groves of Meghalaya revealed the presence of 514 species representing 340 genera and 131 families. In Sikkim, sacred groves are attached to Buddhist monasteries and are called as Gampa forest areas and are managed by lamas. Around 56 sacred groves have been reported in Sikkim. and it's present in highlands of Sikkim, and any human activity here is believed to spell disaster to the region. In Assam, the Bodo and Rabha tribes have the tradition of maintaining sacred groves called as Than. Another tribal group called as Dimasa tribe also maintains sacred grove called as Madaiko or Dikos. Sacred groves are also conserved and managed by Vaishnav monasteries like Shankardev Mats, distributed in different parts of the state, in the state of Assam. In Arunachal Pradesh, the sacred groves are called as Gampa forest areas and are attached to Buddhist monasteries and managed by lamas. 58 of such groves have been reported in Arunachal Pradesh. In Manipur, the Gangte tribes have the tradition of shifting cultivation, and the sacred groves are the only place where they do not follow shifting cultivation. Now, what is shifting cultivation? Tribals move to a virgin forest area, they slash and burn the area and start cultivating agriculture crops. And they repeatedly go on cultivating agriculture crops, and once the fertility status deteriorates, they move to a different area and repeat the process again. They start the process again. This is called a shifting cultivation. And sacred groves, wherever sacred groves are present, the Gangte tribe do not follow shifting cultivation. They also have small bamboo reserves called as mohawk, and extraction of bamboo shoots from this mohawk is totally prohibited. Another type of sacred groves called as Umanglai sacred grove is also present in the state of Manipur. Umanglai is a jungle deity. Around 365 Umanglai sacred groves have been reported in Manipur, and most of the plant species have got medicinal properties. There's a saying in Sanskrit, Nasti Moolam Anaushadham. Nasti Moolam Anaushadham. It means that there are no plants without medicinal properties. Here in this Umanglai sacred groves, 96% of the species are used as medicines. In Jammu and Kashmir, around 150 sacred groves have been reported. They are called as bunnies, and the area varies from around 0.5 to 1,400 acres. And in larger groves, normal forestry activities are carried out, and in smaller groves, complete protection is given, but no systematic studies have been conducted in the sacred groves of Jammu and Kashmir. In the hilly terrain of sacred groves, Himachal Pradesh, many sacred groves are present. Strict protection measures have been provided in the sacred groves of Himachal Pradesh. For instance, the villagers who pass through the groves used to dust their clothes to make sure that they do not carry anything belonging to the grove. Due to such protection measures, climax vegetation representing climax vegetation is being reported in Himachal Pradesh. The dry tract of Rajasthan, many sacred groves are present. Around 1,100 major sacred groves, they are called as orans, are reported in Rajasthan, covering an area of more than 1 lakh hectares. And the Badria Oran, present in the Jaisalmer district bordering Pakistan, is around 15,000 hectares. Its large sacred groves are present in Rajasthan. Madhya Pradesh is a tribal-dominated state, and many sacred groves are present in Madhya Pradesh. The tribals have immense knowledge of the medicinal uses of these plants, and they believe that these plants are not maintained properly or destroyed, natural calamity will ruin their plants. The sacred groves of Madhya Pradesh are called as sarnas, and of late it's been opened up for extraction of non-timber forest produce. In Jharkhand state, sacred groves are called as Jahirdhan or sarnas. And when sal trees come into flowering, sal is one of the primary timbers of India. The primary timbers are teak, sal and devadar are the primary timbers of India. When sal trees come into flowering, they celebrate a festival called as Sahul Sarna in Chotanagpur area in Jharkhand. Around 400 sacred groves have been reported in Maharashtra, and felling a timber and killing of the animal is considered as a taboo in Maharashtra, but the local community can collect twigs, leaf litter, fruits and non-timber forest produced from the sacred groves, and the topsoil from the groves is used as manure to cultivate many agriculture crops. In West Bengal, around 670 sacred groves have been reported. Bamboo sacred groves are also been reported in West Bengal, and no biomass is harvested from the sacred groves of West Bengal. According to a study by Worldwide Fund for Nature, there are 100 important sacred groves in Andhra Pradesh out of 800 sacred groves. Of late, many temples associated with the sacred groves have been modernized by removing the vegetation of the sacred groves. Sacred groves representing mangrove vegetation has been reported from the state of Goa, and many unique mangrove species like Soneraceae, Pseuda, Eurochondra have been reported from the sacred groves of Goa. In Gujarat state, around 42 sacred groves have been documented. 90 species belonging to 51 families have been reported, and cutting or climbing of the trees or removal of the wood is strictly prohibited. And in Gujarat state, the vegetation in the groves is highly varied, including mangroves and freshwater swamps. In Karnataka, the sacred groves are called as Devarakada. Around 1,200 Devarakadas have been reported from Karnataka. And in smaller groves, complete protection is given, and the larger groves, normal forestry activities are carried out. And the local people, the villagers can gather dead wood, non-wood forest produce like mango, jackfruit, and also can tap toddy from caryota urines. In Tamil Nadu, around 450 sacred groves have been reported, and under Stalariksha concept also many trees have been protected in Tamil Nadu. In Tamil Nadu, eastern district without much forest, I got more sacred groves than the western district in Tamil Nadu. This is the Stalariksha concept where the tree is being protected under Stalariksha concept. And Mahua tree worship is being carried out by the tribals of Jharkhand, Chhattisgarh, Odisha, Maharashtra, Madhya Pradesh, and Bihar. And before the onset of the flowering season, the tribals offer prayers and love to the trees, and the tribals collect Mahua flowers, fruits, seeds, and leaves. In Kerala, the sacred groves are called as Kava. There are different types of Kava in Kerala: Sarpa Kava, Ayyappan Kava, Bhagavati Kava, and Yakshi Kava. In Kerala, around 500 hectares of area have been reported in under sacred groves, and the important sacred groves number has been reported as 2,000, and the floristic diversity indices of these groves are equal or nearly equal to the forests of the Western Ghats. The scientific study of the sacred groves was started by two scientists called Gadkil and Vartak in 1975. 4 threatened species, Buchanania, Syzygium travancoricum, Blepharistomma, Turosperma, have been rediscovered from the sacred groves of Kerala. And this is a new genus and a new species discovered from the sacred grove, Kunstleria keralensis. This is a climbing legume. It's a new genus and new species discovered from the sacred groves, not from the forest areas. According to a study by Balasubramanian and Indijudan, 761 important sacred groves are present in Kerala, and the biodiversity potential of these sacred groves were found to be very good when compared to the well-protected evergreen forests of the Western Ghats. According to a study by the Tropical Botanical Garden Research Institute, Palod, 65% of the plant species and tree species belonging to the rare category in sacred grove. That shows the biodiversity potential and the biodiversity richness and importance of the sacred groves. According to a study by our director, Dr. C. Kunjukannan, he has reported 139 important species of flowering plants from a sacred grove called as Karakakava in North Malabar, and many endangered and endemic species have been reported from the sacred grove called Karakakava in North Malabar. Sacred swamps are also present. Myristica swamps are there, Heliocarpus swamps are there, Hatlus are there. And Myristica swamps are dominated by Myristicaceae members, and these relic swamp forests were first discovered in Travancore region in 1960, and many plant and tree species have been discovered from the sacred groves. The Myristica swamp sacred groves, important species are Syzygium travancoricum, Gymnocranthera camerica, Myristica fatua, and Semicarpus catholicanaensis. Catholicans are sacred groves present in Karnataka. Semicarpus catholicanaensis is a critically endangered tree species. They are present in the Katholican forests, Katholican sacred groves of Karnataka. Heliocarpus swamps are also present in Agastyamalai Biosphere Reserve and nearby regions, and only a very few sustainable populations of these Heliocarpaceae trees have been recorded in India because they got very low germination rate. Hudloos are swampy lowlands filled with tall reeds and marsh-dwelling grasses, and they are very important in conserving the ungulate biodiversity in many national parks and tiger reserves of Karnataka, as they remain green even during dry season, and very little is known about the ecological significance of the hudloos of Karnataka. Sacred groves representing unique southern freshwater swamp forest ecosystem has been reported in Thrissur district, in the district of Thrissur, and the predominant species are Syzygium mundagam and Syzygium travancoricum, and around 70% of the tree species, around 80% of the tree species and plant species have got medicinal uses also. Sacred groves representing a unique and understudied tropical dry evergreen forest ecosystem exist in Tamil Nadu, and lianas are the major component of these unique ecosystems, and around 6,000 individuals, so these woody climbers. Lianas are nothing but woody climbers, and woody climbers belonging to 42 species have been recorded from 10 sacred groves in Tamil Nadu. In Karnataka, a study conducted revealed that the conserved sacred groves were comparable to that of the reserve forest. And out of the 25 threatened species, they found that 5 species regenerated only in the sacred groves. Another study revealed that the density of regenerating medicinal plants in the sacred groves were double than that of the natural forest in Karnataka. Medicinal plants regeneration is double in sacred groves when compared to the natural forests. In Cherrapunji, climax vegetation is represented in the form of sacred groves in Cherrapunji region in Meghalaya. We have conducted a case study. Alappuzha is the smallest district in Kerala, and it's called as Venice of the East, and Kuttanad region is one of the places in the world where farming is done below sea level. Another place where farming is done below sea level is Netherlands, the Holland. Alappuzha is the smallest state, as I've mentioned, and this is no sacred. This is one district, this is only a district in Kerala without natural forests. And recently around 13.5 acres have been declared as forest in Alappuzha district, but many sacred groves are present. Small pockets of sacred groves are present in this district. And why should we undertake such studies? The practical relevance of these studies are: studies on floristic diversity of the groves will be of great help in selecting suitable species for each locality, and assessment of these groves will be an excellent tool for any regional biodiversity planning. And the study also will reveal the potentiality of the soil to support various plant species. And we could observe considerable variations with respect to extent of these groves and species composition, and the area of the grove varied from 0.25 cents to 36 acres. 1 cent is 40 meters square, and the area varied from 0.25 cents to 36 acres, and most of the sacred groves were associated with sacred ponds acting as micro watersheds. And this is a pond associated with a sacred grove in Cherthala Taluk in Alappuzha, and they take drinking water. It yields potable water. And the current pandemic is mainly due to deforestation and forest fragmentation, and also due to uncontrolled eating habits. And this is a zoonotic disease, as you know, that this is spread from animals to human beings. And this is a sacred pond associated with a sacred grove in Cherthala Taluk. And this is a temple in Cherthala Taluk. It's called as Cherthala Katyayani temple. It's also called as Kori Ambalam. Fowl worship is done here. People offer fowls to this temple, and these fowls move around the township, and no one converts them into chili chicken or butter chicken, and it's been preserved. Soulatry is practiced here. That's a difference in culture between India and other different countries. And we could enumerate 1,128 sacred groves from the smallest district in Kerala, Alappuzha, covering an area of around 84 hectares. and nearly 40% of the groves had sacred prawns associated with it, and most of the groves are about of many endemic and rare plant species, and we could record 687 plant species belonging to 493 genera and 127 families. And then what's important for this study is that the importance lies here. And, Silent Valley National Park, having an area of around 90 square kilometer core area, 960 species of angiosperms have been reported from 90 square kilometer area of Silent Valley. Whereas from a small area, around 0.84 hectare, less than 1 square kilometer area of sacred groves in Alapura district, we could record 687 plant species. That shows the biodiversity potential of the sacred groves of Alapura district. And I'll introduce you to some of the plant species and tree species present in the sacred groves of Alapura and Kerala. And this is Alstonia scolaris, Edilampali, Alstonia scolaris, and it's believed that it's an abode of spirits, and that's why the tree is being protected. You know why the name Alstonia scolaris has come? The wood of this tree species was being used for making blackboards, and blackboards are used by scholars, and hence the name Alstonia scolaris. And this is Mimicillon species. It's believed that this is a color of Lord Krishna, Krishnavarnam, and the Mimicillon species, Kashava or Kayambu, flowers of Kashava, Mimicillon species, and the fruits of Mimicillon species. And this is Calamus rotang, Calamus species, Chural, Calamus species, and the fruits of Calamus species. Natural regeneration of Calamus in the sacred groves of Alapura and Kerala. And this is called as Uvarian arum, Narumpanal, Uvarian arum. It's a medicinal plant, Uvarian arum. It's a medicinal plant. And this is Myristica malabarica, another medicinal tree, Myristica malabarica. It's a wild relative of Myristica fragrans. This is Myristica fragrans, and this is Myristica malabarica. And this is a sacred grove harboring Myristica malabarica in Alapura. And this is the state flower of Tamil Nadu, Gloriosa superba, Syngrandal, and the seeds of this species of bees being the seeds are being used for making colchicine, a chemical, and it's been cultivated widely in different parts of Tamil Nadu. Plantations of Gloriosa superba, another view of plantation of Gloriosa superba, Syngrandal, the state flower of Tamil Nadu. And the fruits of Syngrandal, says Gloriosa superba. It's being used for making, the seeds are used for making colchicine. And this is Mucuna pruriens, another medicinal plant, Mucuna pruriens. And Naikurna, this is called as Mucuna gigantea. It's called as a tribal pulse also. The seeds contain around 40% proteins. It's called a tribal protein. Mucuna gigantea, Kakavalli, the inflorescence of Mucuna gigantea. And this is Cerbera odalam, another plant, another tree species, Cerbera odalam, which is a poisonous tree species. The fruits of Cerbera odalam, and this is a gymnosperm, Neatomedule, this is a gymnosperm, and female inflorescence of Neatomedule. And this is a male inflorescence of Neatomedule. And this is called as Mathuka nerifolia, Attilipai, Mathuka nerifolia, the young inflorescence, the young foliage of Mathuka nerifolia. This is very beautiful. And it's believed that this tree is, that the goddess Bhagavati resides in this tree, and nobody cuts this tree species, Mathuka nerifolia. And another tree species called as Kavilipai, Mathuka diplostimon, has been rediscovered after 185 years from a sacred grove in Kollam district in Kerala. Kavilipai. This was a study of the Manonmaniam Sundarnath University, and Kavilipai, a tree species, Mathuka diplostimon, rediscovered after a period of 185 years. That shows the biodiversity potential of the sacred groves. And this is Kavilipai, Mathuka diplostimon. And this is Heliocarpus serratus, a wild relative of Rudraksham. Heliocarpus serratus. It yields edible fruits, and many birds are attracted to this tree species when it is in fruiting stage. And this is called as Scaevola scandens, Bhadraksham, Scaevola scandens. It's present in many temples. And this is Cansgera reedi, a medicinal plant. Cansgera reedi. This is called as Abrus precatorius. Abrus precatorius, a medicinal plant. This is the state flower of Kerala, Cassia fistula. Cassia fistula. And this is called as Calophyllum calaba. There are two species of Calophyllum. Calophyllum inophyllum punnai and Calophyllum calaba churu punnai. And the fruits are very small fruits, and it's used for, it yields biodiesel. It's a biodiesel yielding tree species. Calophyllum calaba. And this is Carelia brachyata vallabham. Carelia brachyata vallabham is a mangrove associate. And this is Diospyros peregrina panachi. Diospyros peregrina. And the fruits were used for making gum. The fruit yields a gum, and the gum was being used for repairing fishermen's net. And also was used for making crackers. And this is called as Dalbergia horrida. Dalbergia horrida. Dalbergia horrida is an endemic liner, woody climber to Western Ghats, endemic to Western Ghats. This is called as Trichosanthus cucumerina. Trichosanthus cucumerina, Cartopatovalum. This is a wild relative of our snake gourd. You see the size of the fruit here. It's very small, and you can compare with that of the normal snake gourd. This is called as Trichosanthus cucumerina. Cartopatovalum, wild relative of snake gourd. This is Smilax ceylanica, another medicinal plant. Smilax ceylanica, Karilanchi. This is Sterculia guttata vacca. Sterculia guttata, the flowers are Sterculia guttata. It produces very beautiful flowers. But it emanates, the time of flowering season, it emanates the smell of rotten flesh. It's very difficult for the people to live around this tree species when it is in flowering season. And this is called as Syzygium Syzygium ceylanicum, wild relative of Syzygium cumini, novel, Syzygium ceylanicum, Poochaparam, and it yields edible fruits, and many birds are attracted. Many birds come to this tree species when it is in fruiting season. This is called as Holigarna arnotiana. This is an ingredient of many medicinal Ayurvedic preparation. Gugulu Tiktam, it's an ingredient of Gugulu Tiktam. And generally when we give antibiotics like penicillin injection and all that, they used to give a test dose. And Ayurvedic doctors, before prescribing this Gugulu Tiktam, they used to tell that, they used to advise the patients to take few test doses in the beginning to begin with, and then later on continue with the full doses, because it has allergic reactions. This Holigarna arnotiana produces allergic skin reactions to many people. Natural regeneration of Holigarna arnotiana, Chere or Chare, and this is Olea dioica, another tree species. This is called as Quassia indica or Samadira indica, Karinyotta, Quassia indica. The wood was being used for making wooden footwear, wooden footwear, and fruits also yields ink. Inflorescence of Quassia indica, Samadira indica. Quassia indica is a new name. This is called as Anamutta cocculus, another woody climber, another liner, another medicinal plant. Anamutta cocculus, Injually, Anamutta cocculus, and this is an exotic tree species. Kauropita guinensis, Nagalingamaram. The flowers looks like the hood of a cobra. That's why it's called as Nagalingamaram, Kauropita guinensis of the Lysithidaceae family. This is an exotic tree species. It's also called as cannonball tree because the fruits look like the cartridges of a cannon. Cannonball tree, Kauropita guinensis. This is an ingredient of Dashamulam, Oroxylum indicum, Palagappayani, Oroxylum indicum, and this is a view of the fruits of Oroxylum indicum, Palagappayani, an ingredient of Dashamula. And this is called as Hypnocarpus pentandra maroti. Hypnocarpus pentandra, and this is an oil-yielding tree species. And our Institute of Forest Genetics and Tree Breeding, Coimbatore, has released biopesticide called as Hyact from the fruits of this tree species. Hypnocarpus pentandra, Marotti. This is Morinda citrifolia, noni. Morinda citrifolia, you might have heard of noni, a health drink available in medical shops. It's been prepared from these fruits of this tree species, Morinda citrifolia, noni. It's present in coastal sacred groves of Kerala. This is Mimisops eleni, maram, Mimisops eleni. It yields fragrant flowers, and the flowers are being used for making expensive fragrances, perfumeries. This is called a Salacia fruticosa, a shrub, a medicinal shrub, Salacia fruticosa, and this is Aprosa cardiosperma vetti. This species also yields edible fruits, and many birds are attracted to this tree species. Aprosa cardiosperma vetti. And this is called as Persia macrantha, Kulamava, Persia macrantha. The bark is being used for making agabattis, and it's also a very good host for lac insect. Persia macrantha. This is Aristolochia indica, another medicinal plant. Aristolochia indica, another medicinal plant. And this is called as Sarcostigma clini, Erimathali. Sarcostigma clini. It was being used for making lamp oil. Present, it's not being used. Previously, it was being used for making lamp oil. Sarcostigma clini, Erimathali, another woody climber. And this is called as Barringtonia racemosa, Samudra Chamba. It's present in the banks of rivers and ponds. And this is called as Aphanamixis polystachya, Chennelli. Aphanamixis polystachya. The fruits and leaves in it is put in water and boiled. It gives a red color. Ingredients are there. Many medicinal ingredients are there. And the water, boiled with its leaves and fruits, is being used to give bath to the ladies after their delivery. And this is called as Syzygium caryophyllatum, Cherunyara, Syzygium caryophyllatum, another wild relative of Syzygium cumini. And this is a critically endangered tree species, Syzygium caryophyllatum, a critically endangered tree species. And this species was present only in one sacred grove, having an area of 5 cents in Alpura district. Only one sacred grove harbored this critically endangered tree species. Therefore, the area of the sacred grove is not a matter at all, because the biodiversity potential is of considerable importance. A 5 cent area having a critically endangered tree species. So if the grove goes, this critically endangered tree species also will vanish from this district. And this is called as Hopia ponga, and an endemic tree species to Western Ghats. It's a timber-yielding tree species, ponga, Hopia ponga. And this is another critically endangered tree species, Vatiria indica, Vella pine, and white dammer is obtained from this tree species, and this is also a very good plywood tree species, Vatiria indica, Vella pine, a critically endangered tree species, another view of Vatiria indica. And this is called as Vitex pinnata, and Cartu Maila. It's being used for wood paneling. And this is called as Hugonia mistax, Modurakanni, Hugonia mistax, climbing, it's a climber, woody climber, it's a medicinal plant, Hugonia mistax. And this is called as Strychnus minor. There are two species of Strychnus, Strychnus maximomica and Strychnus minor. From Strychnus maximomica, two alkaloids are being extracted. Strychnine and brucin are two alkaloids which were used for mercy killing, for euthanasia. Previously, at present, it's not being used, and this is called as Strychnus minor. Vallikanjaram, by Strychnus minor, Vallikanjaram, and Strychnus minor. And this is a grove with lot of linas, Vallikava, in Chenganur region of Alapura district. And this is called as Garcinia xanthochymus. It's another wild relative of a cultivated species, Garcinia gummicata. And this is called as Garcinia xanthochymus, a wild relative of Garcinia gummicata. edible fruits are there, and Garcinia xanthochymus, Mundan Puli, and this is called as Lophopetalum whitianum, Vingotta, Lophopetalum whitianum, it yields timber, and this is called as Cinnamomum alabatrum, varana, it's another medicinal plant, Cinnamomum alabatrum, and this is a new genus and new species discovered from the sacred groves, Conslader carolensis, a new genus and new species. And apart from not only Hindus, Muslims also maintain sacred groves, and this is a sacred grove maintained by a Muslim community there. And under Stalaviksha concept also, many sacred trees are being protected by Muslim community in different parts of Kerala. And this is a temple, in southern part of Kerala, Mannarshala temple. It's a very famous temple with around 36 acres of sacred groves present in this temple, and view of the temple. And many exotic tree species are also present in the sacred groves of Mannarshala Grove. Casuarina, you can see Casuarina equisetifolia and Cavaropita gainensis there, and a sacred pond associated with the sacred grove of Mannarshala, and the view inside. And of late, you can see that Mycania micrantha invasion, an exotic weed, Vaira or Mycania micrantha or Dhritarashtra Pache, is being is invading the sacred groves and destroying the sacred groves to a great extent. It covers a canopy and kills the trees. Mycania micrantha invasion is a threat to the sacred groves of southern part of Kerala, and it doesn't even leave the electric poles. This is Mycania micrantha, a noxious weed, and these are other threats to sacred groves. And Eupatorium, Chromalina odorata, another threat to the sacred groves, and Lantana camera. It's also a threat to the sacred groves. It's affecting the natural regeneration of sacred grove. This is another sacred grove of southern part of Kerala inside the sacred. A lot of lianas are present, a lot of climbers are present. This is Acacia caecia, Inja, Acacia caecia invasion. A lot of climbers are there. And this is a sacred grove in the midst of a township in Ernakulam district, and it's called as Iringol Kava. It's around 50 acres of land is there dedicated to sacred groves here. Lot of natural regeneration, plenty of natural regeneration we could observe in the sacred grove, and many woody climbers. Lionesses are present in the sacred grove, many woody climbers, and Mysticaceae members are present, and the sacred pond of the Iringol Kava. And this is Ashoka tree, and Ashoka tree is there, Ashoka flower. Saraka indica trees are there in plenty. The flowers are Saraka indica, Saraka Ashoka. Saraka Ashoka, not Saraka indica. Saraka Ashoka is a new name, and it's an ingredient of Ashoka Arishtam, and it's being used. It's being used for menstrual problems of ladies, women, and Ashoka Arishtam, and of late it's been adulterated with polyalthia because of the lack of availability of Ashoka tree, Saraka Ashoka tree. It's been adulterated with polyalthia tree, and many people do not plant this tree species at the homes because it's believed that when Sita Devi was abducted from India to Sri Lanka during Ramayana, and she was sitting in Ashoka Vanam, and that's why people believe that if you plant Ashoka tree, it brings sorrow. And it's not like that. And Sita Devi was sitting under the tree called Amhersia nobilis. This is a tree, Shimshiba tree. This is Shimshiba tree, Amhersia nobilis. And the dead and fallen wood in the sacred groves of sacred grove of Iringol is being allowed to disintegrate within the sacred grove itself, allowing natural regeneration. And inside the sacred grove, and a board is put by the social forestry department there. Sacred grove, Iringol Kav in the night, during evening time, and we could see littering mainly towards outskirts of the grove. We could see littering in the grove. Littering was found, and this is a sacred grove representing tropical dry evergreen forest in Tamil Nadu. and this is Pudupet Sacred Grove in Tamil Nadu. And you can see exuberance of youth also in the Sacred Grove. Many tourists visit the Sacred Grove and they litter the Sacred Grove to a great extent, and heavy littering was found affecting the natural regeneration of the tree species. And on the other side, in Kerala, the litter is being removed forcefully, and this also is affecting the natural regeneration. It's forceful removal of the litter affecting the natural regeneration of the tree species. And you can see the natural regeneration of a critically endangered tree species, Vatiri indica. This is from our own Sacred Grove in Kerala. My home at Kerala, my ancestral home at Kerala, we maintain 3 sacred groves having an area of around 1 acre, and around 1 acre, and 2 sacred ponds are also being maintained by our family. And this is a natural regeneration noticed in the Sacred Grove of Haripad, Vatiri indica regeneration. And this is a retrogressing Sacred Grove in Alappuzha district, and you can see grazing with big animals also is being found there. And these are island Sacred Groves in Kuttanad region, where farming is done below sea level. And Kuttanad is around 0.5 to 2.5 meters below the sea level. Island Sacred Groves in Kuttanad region, and he is an youngster of a joint family maintaining around 60 Sacred Groves in Kerala. And you could see his fingers, you could see his hands. It's called ectrodactyly, jointed fingers. It's called ectrodactyly, jointed fingers. This is a hand of an elderly person, and around 60% of the family members have got this deformation called ectrodactyly. This is a skeletal aberration. They belong to the blacksmith's family, but they feel that they do not have any problem in carrying out their own activities, and this is ectrodactyly. And due to such beliefs, they believe that it's a gift of God, and due to that belief, they maintain Sacred Groves. It's an abode of snakes, many reptiles are present in the Sacred Grove. And this Sacred Grove, just 1 day before our visit, this was destroyed for canes worth 150 rupees. Just for this 150 rupees, this beautiful Sacred Grove was destroyed. Once it is destroyed, there is no comeback for the Sacred Grove. And when we visit the Sacred Grove, we cannot have no list of Sacred Groves are present. We have to ask a person where is the next Sacred Grove, and when we visit the Sacred Grove, this is the condition to say the vegetation is gone and only idols are present. What happens is that when a person receives a portion of land as an ancestral property from his parents with the Sacred Grove within that, he cannot go and stay inside the Sacred Grove. What he does is that he consults an astrologer and conducts expensive spiritual rituals like this, and they transfer the deity to these idols, and after conducting expensive rituals, and once the deity is being transferred to the idols and it's no longer sacred, and they remove this vegetation, and the vegetation is gone, and only deities are present, and only deities are there. The vegetation is gone, and they put the names also of the deities there, and here also deities are there, only there, the vegetation is gone. And the Sanskritization also, many Sacred Groves have been destroyed. Sanskritization is the process of building temples by removing the vegetation, and due to Sanskritization also, many Sacred Groves have been destroyed, and the list of gods have been provided, but the valuable trees and plants are gone, and the Sacred Groves are destroyed in the most sacred ways. Sacred Groves are destroyed in the most sacred ways like this, after expensive spiritual rituals. At the same time, the stand vegetation is also being maintained in many Sacred Groves. The board in Malayalam says that only during puja time you are allowed to enter into Sacred Groves, otherwise you are not allowed to enter into the Sacred Grove. And this is a mansion, it's an ancient mansion having maintaining around 4 Sacred Groves, around 200 years of old Sacred Groves. And you can see a small protection measures by way of fencing, where a small, simple fencing is giving rise to. It's giving rise to the natural regeneration of many valuable medicinal plants and tree species. And you can see the medicinal, you can see the sacred pond associated with the sacred grove, and you can see the bottom of the sacred pond is also visible. Such a crystal clear water is present in the sacred pond and the sacred grove. And what happens is that he has transferred all the deities to the idols here, and the vegetation has been removed, and this beautiful sacred pond has been reduced in the form of a well here, and he is afraid to remove this tree species. This is Alstonia scalaris tree species, and it's an abode spirit, and he is afraid to remove that, but all of the vegetation has been removed, and he is praying for posterity. Thank you very much for a patient listening.
Thank you so much, sir, for
Most
the
welcome.
excellent speech on sacred groves.
Most welcome, ma'am.
Yes, sir. Yes, sir. Thank you, sir. Dear participants,
Thank you so much for your informative session, sir. The forum is open for discussion.
participants, if you have any questions, you can either post it in the chat box or you can unmute yourself and ask. I think there are no questions.
Okay.
Yeah. Yes, thank you so much, sir.
Thank you so much for the opportunity provided, ma'am. On behalf of the director, we thank Avinashilingam University for having given me the opportunity.
Yes, sir. Thank you, sir. Actually, we are actually overwhelmed to hear your speech, sir. After a long time, we are hearing you.
Thank you, ma'am. Namaste, ma'am. Namaste.
Namaste. Thank you so much, Dr. Kannan.
Thank you.
I was listening to your lecture. I couldn't wish you earlier. It was very nice. Thank you so much for accepting our invitation and coming here. We know you're busy.
Most welcome, ma'am.
Thank you so much.
Most welcome, ma'am. I'm leaving the meeting.
Can we wind up the session, ma'am?
Sir, leave aitara?
Sir, velila pettaru, ma'am.
Okay. It's just a thank you. It's all right. We can.
Participants, tomorrow, it's a Saturday. It is the last day of the UGC Stripe training program. Please join us by the same time, 10:15 a.m. And after the completion, you will be given the final feedback link with which you will get your certificates.
Shall we wind up the session here?
Yes, ma'am.
Thank you, one and all, for your
Gayatri, tomorrow last session at what time?
2:30 to 3:30, ma'am.
Okay.
Ma 3:30 tama session hai mujhe Gayatri.
Yes, ma'am. 2:30 to 3:30 last session hai, ma'am.
Mujhe na 3:30 ki validation.
Yes, ma'am.
3:30 to 4 kila validation mudchilla.
Yes, ma'am. So we'd like all the participants to join us for all the sessions tomorrow, as well as for the valediction after the last session.
The feedback link for today's session is posted in the chat box. Please fill the feedback form. Thank you, one and all, for your patient listening. Today's session is over. Please join by 10:30 tomorrow. Thank you, one and all.